In this video, we'll be looking at homeostasis and response, inheritance, variation and evolution, and finally, ecology. In this lesson, we'll begin by examining what homeostasis is and why it's essential for our survival. Then look at how automatic control systems in the body maintain this stability, explore the concept of negative feedback as the key mechanism behind homeostasis, and finally introduce some examples of homeostatic systems in the human body. Homeostasis comes from Greek words meaning staying the same. It refers to the process of maintaining a stable internal environment within our bodies. The conditions inside your body need to be kept steady, even when the external environment changes. This stability is crucial for survival. Your cells need the right conditions to function properly. This includes the right conditions for enzyme activity, which we've discussed in previous episodes. It involves maintaining a stable internal environment in response to changes in both internal and external conditions. Our bodies have several automatic control systems that regulate our internal environment. These include both nervous and hormonal communication systems. For example, there are control systems that maintain your body temperature, blood glucose level, and water content. All your automatic control systems are made up of three main components that work together to maintain a steady condition. The first component is the receptor. Receptors detect stimuli. They monitor levels of substances or conditions in your body. The second component is the coordination center. This receives and processes the information from the receptors, then organizes a response. The third component is the effector. Effectors produce a response that counteracts any change and helps restore the optimum level. Your automatic control systems keep your internal environment stable using a mechanism called negative feedback. When the level of something like water or temperature gets too high or too low, your body uses negative feedback to bring it back to normal. Let's look at how this works when levels are too high. First, the receptor detects the stimulus, that the level is too high. Next, the coordination center receives and processes this information, then organizes a response. Then the effector produces a response which counteracts the change and restores the optimum level. The level decreases. It's important to understand that the effectors will continue producing responses as long as they're stimulated by the coordination center. This might cause the level to become too much in the opposite direction. Luckily, the receptor detects if the level becomes too different and triggers the negative feedback loop again. This process happens without you thinking about it. It's completely automatic. Your body is constantly making these adjustments to maintain homeostasis. Examples of homeostatic systems in the human body include regulation of body temperature, blood glucose levels, and water content. We'll explore each of these systems in detail in upcoming episodes, looking at the specific receptors, coordination centers, and effectors involved. Understanding homeostasis helps us appreciate how our bodies maintain the delicate balance needed for survival, despite constantly changing internal and external conditions. In this episode, we'll start by examining the basic function of the nervous system and why it's so important, then look at the different parts that make it up, explore how these components work together to coordinate responses, and finally investigate reflex actions and how they help protect us from harm. All organisms need to respond to stimuli, changes in their environment, in order to survive. Single-celled organisms can simply respond directly to their environment, but in multicellular organisms like humans, cells need to communicate with each other first. As organisms evolved and became more complex, they developed nervous and hormonal communication systems. The nervous system is what allows humans to react to their surroundings and coordinate their behavior. It detects stimuli, processes information, and coordinates responses. Let's look at the different parts that make up this complex system. The nervous system can be divided into two main parts. First is the central nervous system, or CNS, which consists of the brain and spinal cord. In vertebrates, animals with backbones, the CNS is protected by bone. The brain is enclosed in the skull, and the spinal cord runs through the vertebral column. The second part is the peripheral nervous system. This consists of sensory neurons and motor neurons that connect the CNS to the rest of the body. Neurons are specialized cells that carry electrical impulses. There are different types of neurons with different functions. Sensory neurons carry information as electrical impulses from receptors to the CNS. Motor neurons carry electrical impulses from the CNS to effectors, the parts of the body that respond to commands from the nervous system. Receptors are cells that detect stimuli. There are many different types of receptors specialized for different stimuli. For example, we have taste receptors on the tongue and sound receptors in the ears. Receptors can form part of larger, complex organs. The retina of the eye is covered in light receptor cells that detect light of different wavelengths. Effectors respond to nervous impulses and bring about a change. Muscles and glands are known as effectors. They respond in different ways. Muscles contract in response to a nervous impulse, whereas glands secrete hormones. The central nervous system acts as a coordination center. It receives information from the receptors and then coordinates a response, which is carried out by effectors. This whole process happens very quickly thanks to the way neurons communicate with each other. Neurons transmit information very quickly to and from the brain, and your brain quickly decides how to respond to a stimulus. The connection between two neurons is called a synapse. Here, the nerve signal is transferred by chemicals which diffuse across the gap. These chemicals then set off a new electrical signal in the next neuron. This allows information to pass from one neuron to the next throughout the nervous system. But some responses happen even more quickly through reflexes. Reflexes are rapid, automatic responses to certain stimuli that don't involve the conscious part of the brain. Reflexes help reduce the chances of being injured. For example, if someone shines a bright light in your eyes, your pupils automatically get smaller so that less light gets into the eye. The passage of information in a reflex from receptor to effector is called a reflex arc. When a stimulus, such as a painful bee sting, is detected by receptors, impulses are sent along a sensory neuron to a relay neuron in the CNS. When the impulses reach a synapse between the sensory neuron and the relay neuron, they trigger chemicals to be released. These chemicals cause impulses to be sent along the relay neuron. When the impulses reach a synapse between the relay neuron and a motor neuron, the same thing happens. Chemicals are released and cause impulses to be sent along the motor neuron. The impulses then travel along the motor neuron to the effector, which is usually a muscle in this example. The muscle then contracts and moves your hand away from the bee. Because you don't have to think about this response, it happens very quickly, helping to minimize damage from harmful stimuli. Understanding the nervous system helps us appreciate how our bodies detect and respond to the world around us. This complex network of cells allows us to react quickly to changes, protecting us from harm and helping us interact with our environment. In our next episode, we'll explore specific parts of the nervous system in more detail, looking at how the brain processes information and coordinates complex responses. Let's begin by going over exactly what we'll cover in this video. First, we'll look at the brain's role as part of the central nervous system. Next, we'll examine different regions of the brain and their specific functions. Then we'll explore the methods scientists use to study the brain. Finally, we'll discuss some of the challenges in investigating and treating brain disorders. Along with the spinal cord, the brain forms part of the central nervous system. It's made up of billions of interconnected neurons, specialized cells that communicate through electrical and chemical signals. The brain is in charge of all our complex behaviors. It controls and coordinates everything we do. Different regions of the brain carry out different functions. Understanding these regions helps us appreciate how this complex organ works. The cerebral cortex is the outer wrinkly layer of the brain. This region is responsible for higher functions like consciousness, intelligence, and language. The cerebellum is located at the back of the brain and is primarily responsible for muscle coordination. It helps ensure our movements are smooth and precise. The medulla is a region that controls unconscious activities, things you don't have to think about doing, like breathing and regulating your heartbeat. Scientists use a range of methods to study the brain and map out which bits do what. One approach involves studying patients with brain damage. If a small part of the brain has been damaged, the effect this has on the patient can tell us a lot about what the damaged part of the brain does. For example, if an area at the back of the brain was damaged by a stroke and the patient went blind, you know that this area has something to do with vision. The brain can also be studied by electrical stimulation. By pushing a tiny electrode into the tissue and giving it a small zap of electricity, scientists can observe what happens. When different parts of the brain tissue are stimulated, it's possible to get an idea of what those parts do. For instance, when a certain part of the brain, known as the motor area, is stimulated, it causes muscle contraction and movement. Magnetic resonance imaging, MRI scanners, are advanced machines that can produce very detailed pictures of the brain structures. Scientists use these to find out what areas of the brain are active when people are doing things like listening to music or trying to remember something. Knowledge of how the brain works has led to the development of treatments for disorders of the nervous system. For example, electrical stimulation of the brain can help reduce muscle tremors caused by nervous system disorders, such as Parkinson's disease. However, the brain is incredibly complex and delicate. This makes the investigation of brain function and the treatment of brain damage or disease challenging. It also carries risks, such as physical damage to the brain during surgery. This means treatments must be developed and applied with extreme caution. Neuroscientists, scientists who study the brain, continue to develop new technologies and methods to better understand this complex organ. Each advancement brings us closer to understanding how the brain works and how to treat neurological disorders more effectively. In our next episode, we'll explore another fascinating sensory organ, the eye, and discover how it allows us to perceive the visual world around us. Let's begin by going over exactly what we'll cover in this video. First, we'll examine the structure of the eye and the function of its various parts. Next, we'll look at how the iris reflex works to protect our eyes in different lighting conditions. Then we'll explore the process of accommodation, how our eyes focus on both near and distant objects. Finally, we'll discuss common vision defects and the ways they can be corrected. The eye is a sophisticated sense organ with several important components working together to enable vision. Let's look at these key structures. The sclera forms the tough, white exterior wall of the eye. This protective layer helps maintain the eye's shape and provides structural support. At the front of the eye sits the cornea, a clear dome-shaped window that begins the process of bending light as it enters the eye. Behind the cornea is the iris, the colorful circular structure that contains muscles. These muscles control the size of the pupil, which is the opening in the center of the iris that allows light to pass through. The lens sits behind the pupil and further directs light toward the back of the eye. It focuses incoming light rays onto the retina, which contains cells that respond to light intensity and different colors. The ciliary muscles and suspensory ligaments work together to adjust the lens shape, allowing us to see objects at varying distances clearly. The optic nerve connects the eye to the brain, transmitting visual information from the light-sensitive cells in the retina to the visual processing centers in the brain. Our eyes have developed sophisticated mechanisms to protect themselves from damage. One important example is the iris reflex, which adjusts our eyes for different light conditions. Intense light can harm the delicate retina, so a protective reflex exists. When excessive brightness is detected by light receptors, a reflex pathway activates that reduces pupil size. This occurs because circular muscles within the iris tighten while the radial muscles loosen. This adjustment limits the amount of light entering the eye, safeguarding the retina from potential damage. In dim environments, the opposite reaction takes place. The radial muscles tighten while the circular muscles relax, causing the pupil to expand. This automatic adjustment optimizes our vision in varying light conditions, allowing more light in when it's dark and restricting light when it's bright. Another crucial function of the eye is focusing on objects at different distances, a process called accommodation. The eye achieves proper focus by altering the lens shape. When viewing nearby objects, a sequence of changes occurs. The eye's ciliary muscles tighten, causing the tension on the suspensory ligaments to decrease. With reduced tension, the naturally elastic lens becomes more curved or rounded. This increased curvature bends light rays more sharply, bringing close objects into focus on the retina. For distant vision, the process reverses. The ciliary muscles loosen, allowing the suspensory ligaments to become taut. This tension pulls on the lens, making it flatter and less curved. The flattened lens bends light less dramatically, allowing distant objects to focus properly on the retina. These adjustments ensure clear vision whether we're reading a book or looking at the horizon. Vision problems develop when the eye cannot properly focus light on the retina. The two most common vision conditions are hyperopia and myopia. People with hyperopia, long-sightedness, can see distant objects clearly but struggle with close-up vision. This happens when the eyeball is shorter than normal or when the lens doesn't curve enough. In these cases, the focal point falls behind the retina rather than directly on it. This creates blurry vision when looking at nearby objects like books or screens. The clinical term for this condition is hyperopia. It can be corrected with convex lenses that curve outward in the middle. These lenses help bend light rays more sharply before they enter the eye, shifting the focal point forward onto the retina. People with myopia, short-sightedness, experience the reverse problem. They see nearby objects clearly but have difficulty with distant vision. This occurs when the eyeball is too elongated or when the lens curves too much. This causes the focal point to fall in front of the retina rather than on it, making distant objects appear blurry. The clinical term for this condition is myopia. It can be addressed using concave lenses that curve inward in the middle. These lenses help spread light rays before they enter the eye, moving the focal point backward onto the retina. Several treatments exist for vision correction beyond traditional eyeglasses. Understanding these options provides important context for those with vision impairments. Contact lenses are small optical devices that rest directly on the eye's surface. They're designed to address specific vision problems by altering how light enters the eye. Many individuals prefer contacts because they're virtually invisible and don't interfere with physical activities. They provide a more natural field of vision compared to glasses. They come in two main varieties: rigid gas permeable lenses and soft lenses. While soft versions offer greater comfort, they present a somewhat higher infection risk than their rigid counterparts. Laser eye correction has become increasingly common for treating vision problems. This surgical approach uses precise laser technology to reshape the corneal tissue. By carefully modifying the cornea's curvature, surgeons can adjust how the eye bends incoming light. This technique can effectively address nearsightedness, farsightedness, and astigmatism. The procedure allows for remarkable precision, often eliminating the need for corrective lenses completely. However, as with any surgical intervention, there are potential risks, including dry eyes, infection, or occasional visual disturbances. For certain patients, particularly those with advanced hyperopia, lens replacement surgery might offer better results than laser treatment. This procedure involves removing the natural lens and replacing it with a custom designed artificial implant made of biocompatible materials. This approach resembles cataract surgery but focuses on vision correction rather than removing clouded lenses. The synthetic lens can be specifically designed to address the patient's unique vision requirements. Understanding how the eye works helps us appreciate the complexity of this remarkable organ. From automatically adjusting to different light levels to focusing on objects at varying distances, the eye performs incredibly sophisticated functions with no conscious effort from us. The ability to correct vision defects has transformed millions of lives, allowing people to see clearly regardless of the natural capabilities of their eyes. Let's begin by going over exactly what we'll cover in this video. First, we'll examine why maintaining a constant body temperature is so important. Next, we'll look at the thermoregulatory center in the brain and how it monitors temperature. Then we'll explore the body's responses to being too hot. Finally, we'll discover the mechanisms that help us warm up when we're too cold. The human body needs to keep its internal temperature at around 37 degrees Celsius. This is the optimum temperature for enzymes in the body to function properly. If body temperature rises or falls too much, enzymes may stop working effectively or even become permanently damaged. This can disrupt vital chemical reactions and potentially be life-threatening. To maintain this precise temperature, the body must balance the energy gained through respiration with the energy lost to the environment. This balance is controlled by the thermoregulatory center in the brain. Located in a region called the hypothalamus, this center contains receptors that are sensitive to the temperature of blood flowing through the brain. The thermoregulatory center also receives information from temperature receptors in the skin. These provide early warning of changes in external temperature before they affect the core body temperature. This system works as a classic example of a negative feedback mechanism. When body temperature deviates from the normal range, responses are triggered that counteract the change. When temperature receptors detect that core body temperature is too high, the thermoregulatory center acts as a coordination center. It processes this information and automatically triggers appropriate effectors. These effectors, such as sweat glands, produce responses that counteract the change in temperature. Similarly, when temperature receptors detect that core body temperature is too low, the thermoregulatory center triggers different effectors, such as muscles, to produce responses that warm the body. Some effectors work antagonistically. One effector heats and another cools, working simultaneously to achieve a very precise temperature. This mechanism allows for a more sensitive response than a simple on-off system. Let's look more closely at the specific responses produced by effectors to counteract changes in body temperature. When you're too hot, several mechanisms activate to help cool you down. First, sweat is produced by sweat glands and evaporates from the skin. This evaporation process transfers energy to the environment, cooling the body. Second, the blood vessels supplying the skin dilate, becoming wider. This is called vasodilation. Vasodilation allows more blood to flow close to the surface of the skin. This helps transfer heat energy from deeper tissues to the skin where it can be lost to the surroundings. These cooling mechanisms are particularly effective when the air is dry, promoting evaporation, and when there is a temperature difference between the body and the environment. When you're too cold, a different set of responses occur to conserve and generate heat. First, the hairs on your skin stand up. This is often called goosebumps and creates a layer of trapped air that acts as insulation. Second, sweat production stops. This prevents heat loss through evaporation. Third, blood vessels supplying skin capillaries constrict, becoming narrower. This is called vasoconstriction. Vasoconstriction reduces blood flow to the skin surface. This minimizes heat loss and helps maintain the temperature of vital internal organs. Fourth, when you're very cold, your muscles contract rapidly and involuntarily in a process called shivering. This increased muscle activity generates heat through respiration, helping to warm the body. These heating and cooling mechanisms work together to maintain our core temperature within a narrow range, despite varying external conditions. Both sets of responses are automatic. We don't consciously control them. They're controlled by the autonomic nervous system, which regulates many unconscious bodily functions. Understanding temperature regulation helps us appreciate the sophisticated control systems in our bodies. This homeostatic mechanism demonstrates how different parts of the body, the brain, skin, blood vessels, and muscles work together to maintain the stable internal conditions necessary for life. Let's begin by going over what we'll cover. First, we'll examine what hormones are and how they function. Next, we'll look at the major endocrine glands and their roles. Finally, we'll compare hormonal and nervous communication. Our bodies need to send information between different parts to coordinate activities. While the nervous system uses electrical impulses traveling along neurons, the endocrine system uses chemical messengers called hormones. Hormones are chemical molecules released directly into the bloodstream. They travel throughout the body but only affect particular cells in specific organs, called target organs. These target cells have special receptors that recognize and respond to specific hormones. Unlike the rapid but short lived signals of the nervous system, hormones typically create slower but longer lasting effects. Hormones are produced by specialized structures called endocrine glands. These glands secrete their hormones directly into the bloodstream rather than through ducts. The pituitary gland, located at the base of the brain, is often called the master gland because it produces many hormones that regulate body conditions and control other glands. It directs them to release hormones that bring about changes throughout the body. The thyroid gland in the neck produces thyroxine, which regulates metabolism, heart rate, and temperature. This hormone controls how quickly the body uses energy. The adrenal glands, situated on top of the kidneys, produce adrenaline. This hormone prepares the body for fight or flight responses during stress or danger. The pancreas produces insulin, which regulates blood glucose levels. We'll explore this crucial hormone in more detail in our next episode. In females, the ovaries produce estrogen, which is involved in the menstrual cycle and secondary sexual characteristics. In males, the testes produce testosterone, which controls puberty and sperm production. While both the nervous and hormonal systems help coordinate body functions, they work quite differently. The nervous system acts very quickly, but for a short time, affecting precise areas of the body. For example, if you touch something hot, nerve signals rapidly travel to and from your brain, causing you to pull away immediately. The endocrine system acts more slowly but has longer lasting effects, influencing the body in a more general way. When you experience stress, adrenaline is released gradually but continues affecting your body for some time. If you're not sure whether a response is nervous or hormonal, consider its timing and duration. Very quick responses are typically nervous, while those lasting longer are usually hormonal. Understanding the endocrine system helps us appreciate how our bodies maintain balance through chemical signals. These hormones ensure our cells work together harmoniously despite changing internal and external conditions. In our next episode, we'll explore one of the most important hormonal systems, insulin and the control of blood glucose. Let's begin by going over what we'll cover. First, we'll examine what affects blood glucose levels in our bodies. Next, we'll look at how insulin and glucagon work together to maintain balance. Finally, we'll explore what happens when this control system doesn't work properly in diabetes. Our blood glucose concentration needs careful regulation. Too much or too little glucose in the bloodstream can cause serious health problems. The pancreas is the key organ responsible for monitoring and controlling blood glucose levels. It produces two important hormones, insulin and glucagon, that work in opposition to keep blood glucose levels stable. Several factors affect the amount of glucose in our blood. When we eat foods containing carbohydrates, these are digested, and glucose enters our bloodstream from the intestines. At the same time, normal cell metabolism constantly removes glucose from the blood as cells use it for energy. Physical activity, especially vigorous exercise, increases the rate at which glucose is removed from the blood, as active muscles require more energy. When we have excess glucose, it can be stored as glycogen in the liver and muscles for later use. The liver can store a significant amount of glycogen, making it a crucial organ for blood glucose regulation. The level of glucose in the blood must be kept steady. The normal range is quite narrow, and deviations can have serious consequences. This is why the body has developed sophisticated mechanisms to monitor and control blood glucose levels. The body uses a negative feedback system to maintain blood glucose within the normal range. This means that any change away from the normal range triggers responses that counteract the change, bringing levels back to normal. When blood glucose levels rise too high, perhaps after eating a meal rich in carbohydrates, the pancreas detects this change and responds by secreting insulin into the bloodstream. Insulin causes liver cells to convert glucose into glycogen for storage. This removes excess glucose from the blood, bringing levels back down to normal. Insulin also helps muscle and fat cells absorb more glucose from the bloodstream, further reducing blood glucose levels. It does this by causing special glucose transporters to move to the cell membrane, allowing glucose to enter the cells. The overall effect of insulin is to reduce blood glucose concentration by moving glucose from the blood into cells, where it can be used or stored. Conversely, when blood glucose levels fall too low, perhaps during exercise or when we haven't eaten for a while, the pancreas detects this change and secretes glucagon. Glucagon has essentially the opposite effect to insulin. It causes liver cells to convert stored glycogen back into glucose, which is released into the bloodstream. This raises blood glucose levels back to normal. These two hormones, insulin and glucagon, work together in a balanced way to maintain steady blood glucose levels, regardless of whether we've just eaten or haven't had food for hours. This is a classic example of antagonistic hormone action in maintaining homeostasis. Unfortunately, this control system can fail, resulting in a condition called diabetes. There are two main types of diabetes, each with different causes but similar effects. In type 1 diabetes, the pancreas cannot produce enough insulin. This is usually caused by an autoimmune reaction where the body's immune system attacks and destroys the insulin producing cells in the pancreas. Without insulin, glucose cannot enter the body's cells and remains in the bloodstream, leading to high blood glucose levels. People with type 1 diabetes need regular insulin injections throughout their lives to control their blood glucose levels. The amount of insulin required depends on several factors, including diet, physical activity levels, and individual metabolism. Modern insulin therapy can be very effective, allowing people with type 1 diabetes to live normal, healthy lives. Type 2 diabetes occurs when body cells become resistant to insulin. The pancreas still produces insulin, sometimes even in larger than normal amounts, but cells don't respond properly to it. This means glucose stays in the blood rather than being taken up by cells, causing high blood glucose levels. Being overweight increases the risk of developing type 2 diabetes, as does lack of regular exercise. Type 2 diabetes often develops gradually, and many people have the condition for years before it's diagnosed. Early symptoms can include increased thirst, frequent urination, fatigue, and blurred vision. Treatment for type 2 diabetes usually involves lifestyle changes, such as improved diet and increased exercise. In more severe cases, medication to increase insulin sensitivity or stimulate insulin production may be needed. Understanding glucose regulation helps us appreciate the complex balancing act our bodies perform constantly. This homeostatic mechanism is critical for maintaining the stable internal environment our cells need to function properly. In our next episode, we'll explore other hormones and their roles in regulating different aspects of body function. Let's begin by going over exactly what we'll cover in this video. First, we'll examine the basic structure and function of the kidneys. Next, we'll look at how the kidneys filter blood and remove waste products. Then we'll explore how water balance is maintained through hormonal control. Finally, we'll investigate what happens when kidneys fail and the treatments available. The kidneys are bean-shaped organs located towards the back of the abdominal cavity, one on each side of the spine. Though relatively small, they perform several vital functions that maintain homeostasis in the body. The primary role of the kidneys is to filter waste products and excess substances from the blood. This cleaning process is essential for removing potentially harmful compounds while retaining useful materials. When blood passes through the kidneys, a complex filtration process occurs. This process is remarkably selective, allowing the kidneys to remove specific substances while returning others to the bloodstream. This filtration happens in microscopic structures called nephrons, which are the functional units of the kidneys. Each kidney contains about 1 million nephrons, giving them tremendous filtering capacity. The first stage of kidney function is ultrafiltration, where small molecules are filtered out of the blood as it passes through tiny blood vessels in the kidneys. The second stage is selective reabsorption, where useful substances are reabsorbed back into the bloodstream. This ensures that valuable materials aren't wasted by being excreted. The substances that aren't reabsorbed are removed from the body in urine, which is stored in the bladder before being expelled. Let's look more closely at the specific waste products removed by the kidneys. The first major waste product is urea. Urea forms when proteins are broken down in the liver. Proteins contain nitrogen, and when excess amino acids are broken down, this nitrogen needs to be removed from the body. Proteins and amino acids can't be stored by the body, unlike fats and carbohydrates. When we consume more protein than needed for growth and repair, the excess amino acids are converted to urea in a process called deamination. Ammonia is actually produced as an initial waste product during this process, and it's highly toxic. The liver quickly converts ammonia to urea, which is much less harmful and can be safely transported in the bloodstream to the kidneys for excretion. The kidneys also regulate iron levels in the blood. ions such as sodium are taken into the body, in food, and then absorbed into the bloodstream. If the ion content of the body is wrong, this could upset the balance between ions and water, making it difficult to draw water into cells by osmosis. Having the wrong amount of water can damage cells or mean they don't work normally. Some ions are lost in sweat, which tastes salty due to its sodium content. However, the amount of ions in the body is primarily regulated by the kidneys. The right amount of ions must be maintained in the blood after filtration, and the rest is removed from the body in urine. Perhaps the most critical function of the kidneys is regulating water balance. Our bodies are approximately 70% water, and maintaining the correct amount is essential for all cellular functions. The body has to constantly balance the water coming in against the water going out. We lose water from the skin through sweat and from the lungs when breathing out. We can't control how much water we lose in these ways, so the amount of water is balanced by the amount we consume and the amount removed by the kidneys in urine. A specific hormone known as antidiuretic hormone regulates how concentrated our urine becomes. The pituitary gland, located at the base of the brain, secretes this hormone into the circulatory system. Special receptors in the brain continuously measure blood water levels, sending signals to the pituitary to adjust ADH secretion as needed. The whole process of water content regulation is controlled by negative feedback. This means that if the water content gets too high or too low, a mechanism will be triggered that brings it back to normal. When water content in the blood is too low, receptors in the brain detect this change. The coordination center in the brain receives this information and coordinates a response. The pituitary gland releases more ADH, so more water is reabsorbed from the kidney tubules back into the blood. This results in a smaller volume of more concentrated urine. Conversely, when water content in the blood is too high, the brain receptors detect this change. The coordination center receives this information and coordinates a different response. The pituitary gland releases less ADH, so less water is reabsorbed from the kidney tubules. This results in a larger volume of more dilute urine, removing the excess water from the body. This precise control mechanism allows our bodies to adapt to varying water intake and environmental conditions while maintaining a stable internal environment. What happens if the kidneys stop working properly? If someone's kidneys fail, waste products build up in their blood, and they lose the ability to control the levels of ions and water in their body. Eventually, this leads to death if not treated. There are two main treatments for kidney failure: regular dialysis or a kidney transplant. Dialysis is a medical procedure that artificially filters the blood. It's designed to keep concentrations of dissolved substances in the blood at normal levels and to remove waste products. In a dialysis machine, the patient's blood flows between partially permeable membranes surrounded by dialysis fluid. This fluid has the same concentration of dissolved ions and glucose as healthy blood. This means these substances won't be lost from the blood during dialysis. However, waste products, such as urea, are absent from the dialysis fluid. This creates a concentration gradient that causes urea and excess ions and water to diffuse across the membrane from the blood into the dialysis fluid. Many patients with kidney failure have to undergo dialysis sessions 3 times a week, with each session taking 3 to 4 hours. It's not a pleasant experience and is expensive to provide, but dialysis can buy patients with kidney failure valuable time until a donor organ is found. The alternative treatment is a kidney transplant, which is currently the only cure for kidney failure. Healthy kidneys for transplant typically come from people who have died suddenly and were on the organ donor register or carried a donor card. Kidneys can also be donated by living people, as we only need one functioning kidney to survive. However, there is a small risk to the person donating the kidney. One challenge with transplants is that the donated kidney can be rejected by the patient's immune system. To prevent this, patients must take immunosuppressant drugs, which reduce the activity of their immune system. While these drugs help prevent rejection, they also make the patient more vulnerable to infections. Despite these challenges, transplants are generally cheaper in the long run than dialysis and put an end to the hours patients would otherwise spend on dialysis machines. Understanding how the kidneys maintain water and nitrogen balance helps us appreciate the complex systems that keep our bodies functioning properly. These remarkable organs work continuously behind the scenes, filtering our blood about 60 times each day and producing about 1 to 2 liters of urine. In our next episode, we'll explore the role of hormones in human reproduction and how they control the development of secondary sexual characteristics and the menstrual cycle. To get started, let me walk you through what we'll be covering. First, we'll examine how hormones trigger the development of secondary sexual characteristics during puberty. Next, we'll look at the main reproductive hormones in males and females. Then we'll explore the menstrual cycle in detail, including its four main stages. Finally, we'll investigate how different hormones control this cycle. At puberty, the body undergoes significant changes as it develops from a child into an adult capable of reproduction. These changes are triggered by sex hormones. In males, the main reproductive hormone is testosterone. This hormone is produced by the testes and stimulates sperm production. Testosterone also triggers the development of secondary sexual characteristics, such as facial hair. In females, the main reproductive hormone is estrogen. It's produced by the ovaries and brings about physical changes, such as breast development. Estrogen is also involved in the menstrual cycle and helps eggs mature. These hormones don't just appear suddenly at puberty. They're actually released in very small amounts throughout childhood, but their levels increase dramatically during the teenage years. The menstrual cycle is a complex series of changes that occurs in a woman's reproductive system. Each cycle takes approximately 28 days, though this can vary between individuals. The cycle involves the release of an egg from a woman's ovaries and the preparation of the uterus to receive a fertilized egg. If fertilization doesn't occur, the uterus lining breaks down and is shed. The menstrual cycle can be divided into four main stages. Stage 1 is menstruation, where the uterus lining breaks down for about 4 days. Stage 2 involves the rebuilding of the uterus lining. From day 4 to day 14, a thick spongy layer full of blood vessels forms, ready to receive a fertilized egg. Stage 3 occurs around day 14, when an egg develops and is released from the ovary. This is called ovulation. In stage 4, the uterus wall is then maintained for about 14 days until day 28. If no fertilized egg has implanted in the uterus wall by this time, the spongy lining starts to break down and the cycle begins again. This cycle is controlled by a complex interplay of four hormones: follicle stimulating hormone, luteinizing hormone, estrogen, and progesterone. Follicle stimulating hormone is produced in the pituitary gland. It causes an egg to mature in one of the ovaries, specifically in a structure called a follicle. Follicle stimulating hormone also stimulates the ovaries to produce estrogen, which in turn causes the lining of the uterus to grow. Estrogen has additional roles in the cycle. It stimulates the release of luteinizing hormone and inhibits the release of follicle stimulating hormone. Luteinizing hormone is also produced by the pituitary gland. It stimulates the release of an egg at day 14 of the cycle, the process known as ovulation. After ovulation, the remains of the follicle develop into a structure called the corpus luteum, which produces progesterone. Progesterone maintains the lining of the uterus during the second half of the cycle. If progesterone levels fall, the lining breaks down, leading to menstruation. Progesterone also inhibits the release of luteinizing hormone and follicle stimulating hormone, preventing further eggs from developing during the current cycle. This entire system works through negative feedback loops. For example, when estrogen levels rise, they inhibit follicle stimulating hormone production, preventing additional eggs from maturing. When progesterone levels rise, they inhibit luteinizing hormone, preventing additional ovulations. If a fertilized egg implants in the uterus wall, it produces hormones that maintain the lining, preventing its breakdown and the start of a new cycle. This is one of the first signs of pregnancy. Understanding these hormonal processes is important not just for appreciating the complexity of human reproduction, but also for developing contraceptive methods and treatments for various reproductive disorders. The precise timing and interaction of these hormones ensure that the reproductive system functions properly, preparing the body each month for the possibility of pregnancy. In our next episode, we'll explore how an understanding of these hormonal mechanisms has led to the development of various contraceptive methods that help people plan their families. Let's start by outlining the main points we'll go over. First, we'll briefly review how pregnancy occurs. Next, we'll examine how hormonal methods of contraception work. Then, we'll look at various barrier methods that prevent sperm and egg meeting. Finally, we'll explore some other approaches to preventing pregnancy. Pregnancy happens when a sperm cell reaches and fertilizes an egg that has been released from the ovary during ovulation. Contraception methods aim to prevent this process in different ways. Hormones can be used as an effective method of contraception. These work by disrupting the normal reproductive cycle we discussed in our previous episode. Estrogen can be used to prevent egg release from the ovaries. This might seem counterintuitive since estrogen naturally helps stimulate egg release. However, when taken continuously in contraceptive form, estrogen actually prevents the production of follicle stimulating hormone. Without follicle stimulating hormone, egg development and maturation stop and ovulation doesn't occur. Progesterone also reduces fertility in several ways. It stimulates the production of thick cervical mucus, which creates a barrier that makes it difficult for sperm to swim through and reach an egg. Additionally, progesterone thins the uterus lining, making it less suitable for a fertilized egg to implant. Many contraceptive methods use combinations of these hormones. The combined oral contraceptive pill contains both estrogen and progesterone. This pill is over 99% effective at preventing pregnancy when used correctly. However, it can cause side effects like headaches and mood changes, and doesn't protect against sexually transmitted diseases. The progesterone only pill has fewer side effects than the combined pill and is just as effective when taken properly. Other hormonal contraceptive methods include the contraceptive patch, which contains estrogen and progesterone. This small patch sticks to the skin, and each one lasts for 1 week. The contraceptive implant is inserted under the skin of the arm. It releases a continuous amount of progesterone over an extended period, stopping egg release and thickening cervical mucus. Contraceptive injections also contain progesterone, and each dose lasts 2 to 3 months. Intrauterine devices are T-shaped devices inserted into the uterus. Some types release hormones like progesterone, while copper intrauterine devices don't contain hormones but still prevent pregnancy by creating an environment hostile to sperm. Non hormonal contraception methods work by creating physical barriers that stop sperm from reaching the egg. Condoms are worn over the penis during intercourse to prevent sperm entering the vagina. There are also female condoms that line the inside of the vagina. Both types of condoms are the only form of contraception that offer protection against sexually transmitted diseases. A diaphragm is a shallow plastic cup that fits over the cervix, forming a barrier. It must be used with spermicide, a substance that disables or kills sperm. Spermicide can also be used alone as a form of contraception, but it's not as reliable as when combined with other methods. There are other approaches to avoiding pregnancy beyond hormonal and barrier methods. Sterilization involves cutting or tying the fallopian tubes in females or the sperm duct in males. This is a permanent procedure with a very small chance that the tubes can rejoin. Some people use natural methods to avoid pregnancy. This involves tracking when the woman is most fertile during her menstrual cycle and avoiding intercourse on those days. While popular with people who prefer not to use hormonal or barrier methods, natural timing is less effective than most other contraception methods. Abstinence, not having sexual intercourse, is the only method that is 100% effective at preventing pregnancy. Each contraceptive method has advantages and disadvantages regarding effectiveness, ease of use, potential side effects, and protection against sexually transmitted diseases. Understanding how different contraceptive methods work allows people to make informed choices about their reproductive health and family planning. In our next episode, we'll explore how hormones can be used to treat infertility when people have children but are experiencing difficulties conceiving naturally. Before we get into the details, here's what's coming up. First, we'll examine how hormones can be used to increase fertility. Next, we'll look at in vitro fertilization and how it works. Then we'll explore the benefits and challenges of fertility treatments. Finally, we'll briefly consider some of the ethical questions surrounding these technologies. Some women experience fertility problems because their hormone levels aren't optimal for conception. For example, they may have levels of follicle stimulating hormone that are too low to trigger egg maturation. When eggs aren't properly released from the ovaries, natural pregnancy becomes difficult or impossible. Fortunately, medical science has developed fertility drugs that contain hormones like follicle stimulating hormone and luteinizing hormone. These can be administered to stimulate ovulation, the release of eggs from the ovaries. These treatments help many women become pregnant who previously couldn't conceive. However, the success is not guaranteed for everyone. Some women may need multiple attempts before achieving pregnancy, which can be expensive and emotionally challenging. Another potential issue is that hormone treatments can sometimes stimulate too many eggs, resulting in multiple pregnancies, such as twins or triplets. For couples who still struggle to conceive using hormone treatments alone, in vitro fertilization, IVF, provides another option. IVF involves collecting eggs from a woman's ovaries and fertilizing them in a laboratory using sperm from her partner or a donor. Before the egg collection procedure, the woman is typically given FSH and LH to stimulate several eggs to mature. This increases the chances of obtaining viable eggs for fertilization. The collected eggs are then combined with sperm in a laboratory dish. If fertilization occurs successfully, the resulting embryos are grown in a special incubator. For couples where the man has a very low sperm count, a technique called intracytoplasmic sperm injection may be used. This involves directly injecting a single sperm into an egg. Once the embryos have developed into small balls of cells, one or more are transferred to the woman's uterus. Any remaining healthy embryos can be frozen for future use if needed. Fertility treatment offers hope to many couples who couldn't otherwise have biological children. For those who have longed for a family, this technology can fulfill a deeply held wish. However, the process comes with challenges. The success rate of IVF is relatively low. This means that many couples face disappointment. The process can also be emotionally stressful, with cycles of hope and potential disappointment. Many find the uncertainty difficult to manage. It's also physically demanding for women who may experience side effects from the hormone treatments, such as abdominal pain, bloating, and headaches. Multiple births are more common with fertility treatments. While many parents are delighted with twins or triplets, multiple pregnancies carry higher risks of complications for both the mother and babies. Advances in laboratory techniques have improved IVF outcomes over the years. Specialized microtools help embryologists handle eggs and sperm under the microscope with greater precision. Time-lapse imaging technology now allows continuous monitoring of embryo development without removing them from the incubator. This helps identify the embryos most likely to result in successful pregnancies. Some people have ethical concerns about certain aspects of IVF treatment. The process often results in unused embryos that may eventually be destroyed, which some find problematic. Genetic testing of embryos before implantation is now possible, but raises questions about selecting embryos based on their genetic characteristics. Despite these concerns, IVF has helped millions of people worldwide become parents who otherwise might not have had the opportunity. Understanding how hormones regulate fertility has been crucial in developing these treatments. The careful application of this knowledge has transformed the lives of many families. In our next episode, we'll explore the complex topic of negative feedback systems and how they help regulate various hormones throughout the body. Let's take a look at what we'll be covering in this video. First, we'll examine adrenaline and its role in preparing the body for fight or flight. Next, we'll look at thyroxine and how it regulates metabolism. Finally, we'll explore how negative feedback systems control hormone levels in the body. Adrenaline is a hormone released by the adrenal glands, which sit just above the kidneys. It's sometimes called the emergency hormone because it prepares your body to respond to stressful or scary situations. When you're faced with danger or stress, your brain detects fear or stress and sends nerve impulses to your adrenal glands, which respond by secreting adrenaline. Adrenaline prepares the body for fight or flight by triggering a series of rapid changes. It increases blood pressure and heart rate, ensuring more blood flows to the muscles and brain. It also increases your breathing rate, which brings more oxygen into the body. Adrenaline raises blood glucose levels to provide extra energy and increases the rate of respiration in cells. Together, these changes prepare you to either face the danger or run away from it, the classic fight or flight response. Now let's turn to thyroxine, which plays a very different role in the body. Thyroxine is released by the thyroid gland located in the neck. Unlike adrenaline, which prepares us for emergencies, thyroxine regulates our day-to-day metabolism, the chemical processes that occur within our cells. Thyroxine helps control the basal metabolic rate, the speed at which chemical reactions happen in the body when at rest. It's important for regulating protein synthesis for growth and development. The hormone also supports many essential processes like stimulating protein synthesis for growth and development. Without adequate thyroxine, normal development cannot occur. Both adrenaline and thyroxine are controlled by negative feedback systems. This is a key mechanism that maintains hormone levels within narrow limits. In a negative feedback system, when levels of a hormone rise too high or fall too low, it triggers responses that bring the level back to normal. Let's look at how this works for thyroxine. The release of thyroxine is controlled by thyroid stimulating hormone, TSH, which is released by the pituitary gland. When the level of thyroxine in the blood is too low, the pituitary gland secretes more TSH. This stimulates the thyroid gland to release more thyroxine, bringing levels back to normal. Conversely, when thyroxine levels are too high, the secretion of TSH from the pituitary gland is reduced. This decreases the amount of thyroxine released from the thyroid, bringing levels back down to normal. This is a classic example of negative feedback. The system responds in the opposite direction to the change, helping maintain balance. Negative feedback is crucial for maintaining homeostasis, keeping conditions in the body relatively constant. Without it, hormone levels could fluctuate wildly, causing serious disruption to bodily functions. Understanding how hormones like adrenaline and thyroxine work and how their levels are regulated helps us appreciate the sophisticated control systems that keep our bodies functioning properly. In our next episode, we'll be exploring plant hormones, discovering how they regulate plant behavior. To start off, here's a quick overview of what we'll go through in this video. First, we'll examine auxin, a key hormone that controls plant growth. Next, we'll look at how auxin helps plants respond to light and gravity. Then we'll explore gibberellins and their role in plant development. Finally, we'll discover how ephine controls fruit ripening and how these hormones are used commercially. Plants don't just grow randomly. Their development is carefully controlled to ensure they grow in the right direction and respond appropriately to environmental conditions. Plant hormones are chemical substances produced in small amounts that influence plant growth and development. They're transported around the plant to coordinate responses to the environment. Auxin is one of the most important plant hormones. It controls growth near the tips of shoots and roots, helping to determine the plant's overall shape. Auxin responds to environmental cues like light and gravity. When a plant detects these stimuli, it adjusts auxin distribution, which changes how different parts of the plant grow. Auxin is produced in the tips of plants and moves backwards to stimulate cell elongation. This process occurs in the cells just behind the tip. If the tip of a shoot is removed, no auxin is available, and the plant may stop growing. This is why pruning can control a plant's size and shape. One interesting feature of auxin is that it affects different plant parts in different ways. Extra auxin promotes growth in the shoot but inhibits growth in the root. This produces a coordinated growth pattern that helps the plant thrive. Let's look at how auxin helps plants respond to light, a process called phototropism. Plants need light for photosynthesis, so it's advantageous for them to grow toward light sources. When a shoot tip is exposed to light, more auxin accumulates in the shade than the side that's in the light. This uneven distribution causes cells on the shaded side to grow longer. As a result, the shoot bends toward the light. This bending allows plants to maximize their exposure to sunlight, which is essential for photosynthesis. Plants also respond to gravity, a process called gravitropism. This helps roots grow downward into the soil and shoots grow upward. When a shoot is growing sideways, gravity produces an unequal distribution of auxin in the tip, with more auxin on the lower side. This causes the lower side to grow faster, bending the shoot upwards. In roots, the opposite happens. A root growing sideways will also have more auxin on its lower side. However, in roots, extra auxin inhibits growth. This means the cells on top elongate faster and the root bends downwards. These responses ensure that shoots grow up toward light, while roots grow down into the soil where they can absorb water and minerals. Scientists have found many practical applications for plant hormones. They can be extracted from plants, or artificial copies can be made. These hormones are then used in agriculture and horticulture. Auxins have several commercial uses. One important application is in weed control. Most weeds in fields or lawns are broad-leaved plants, unlike crops and grasses which have narrow leaves. Selective weed killers have been developed using auxins, which only affect the broad-leaved plants. They disrupt the normal growth patterns of these weeds, causing them to die while leaving grasses and crops unharmed. Auxins are also used as rooting powders. A cutting is a part of a plant that has been cut off, like the end of a branch, with a few leaves on it. Normally, these cuttings would not grow roots. However, if rooting powder containing auxins is applied, cuttings can produce roots rapidly and start growing as new plants. This enables growers to create many identical copies of a valuable plant much more quickly than growing from seeds. Another application is in tissue culture, where plants are grown from small clusters of cells. Hormones, including auxins, are added to the growth medium along with nutrients to stimulate the cells to divide and form both roots and shoots. Gibberellin is another important plant growth hormone. It stimulates seed germination, stem growth, and flowering. One major use of gibberellins is controlling seed dormancy. Many seeds won't germinate until they've been through certain conditions, such as a period of cold or dryness. This is called dormancy and helps ensure seeds germinate at the right time of year. Seeds can be treated with gibberellins to break dormancy and make them germinate at times of year when they wouldn't normally grow. This gives farmers more control over when crops develop. Gibberellins also help with flowering. Some plants require specific conditions to flower, such as longer days or low temperatures. When treated with gibberellins, they can flower without these environmental triggers, which is useful for greenhouse growers who want to produce flowers all year round. Another commercial application is increasing fruit size. Seedless varieties of fruit often don't grow as large as seeded fruit. By applying gibberellins to these fruits, they grow larger to match the size of normal types, making them more appealing to consumers. Ethene is a gas produced by aging parts of plants. It plays a crucial role in fruit ripening by controlling cell division and stimulating enzymes that cause fruit to ripen. Commercially, ethene is used to speed up the ripening of fruits. Fruits are often harvested while they're still green, so they don't get damaged during transport. The gas is then added to ripening rooms at the destination to trigger the ripening process just before sale. This means that fruit can be picked, transported, and arrive at supermarkets in perfect condition. Without this technique, much more fruit would be damaged during shipping. Ripening can also be delayed by storing fruit in conditions that block ethene's effect or reduce the amount of ethene the fruit can produce. This extends the shelf life of many fruits. Understanding plant hormones has revolutionized agriculture and horticulture. By controlling these natural chemical messengers, we can influence how plants grow, when they flower, and how quickly fruits ripen. This knowledge has helped increase crop yields, reduce waste, and develop new methods of plant propagation. As research continues, we'll likely discover even more ways to work with plant hormones to improve food production and create more resilient crops. Our next episode will go into the topic of sexual and asexual reproduction. Before we dive in, let's outline the key topics we'll discuss. First, we'll examine the key differences between sexual and asexual reproduction. Next, we'll look at how each method works and the types of offspring they produce. Then we'll explore the advantages and disadvantages of both reproductive strategies. Finally, we'll discover how some organisms can switch between both methods depending on their circumstances. Living organisms can reproduce in two fundamentally different ways. Let's first look at sexual reproduction, which involves two parents combining their genetic material. In sexual reproduction, genetic information from two organisms, a father and a mother, is combined to produce offspring that are genetically different from either parent. The male and female parents produce specialized reproductive cells called gametes. In animals, these are sperm cells from males and egg cells from females. In humans, each gamete contains 23 chromosomes, exactly half the number found in normal body cells. This reduction is vital to maintain the correct chromosome number across generations. During fertilization, the egg and sperm fuse together, forming a cell with the full number of chromosomes, half from each parent. This is why offspring inherit characteristics from both parents. The specific mixture of chromosomes they receive determines many of their traits. This blending of genetic information creates a variation among offspring. Siblings often look different because they inherit different combinations of genes from their parents. Sexual reproduction isn't limited to animals. Flowering plants also reproduce sexually. They produce male gametes in pollen and female gametes in the ovules of their flowers. When pollen reaches the female part of a flower, fertilization can occur, leading to seed formation. In contrast, asexual reproduction involves only one parent and the offspring are genetically identical to that parent. Asexual reproduction happens through mitosis, a cell division process where one cell makes two genetically identical copies. The new cells contain exactly the same genetic information as the parent cell. Since there's only one parent, no fusion of gametes, and no mixing of chromosomes, the offspring are clones of the parent, genetically identical in every way. Many organisms reproduce asexually, including bacteria, some plants, and certain animals. For example, bacteria simply divide in two, with each new cell being a clone of the original. Now that we understand the basic differences, let's consider why these two strategies exist. Each has distinct advantages that make it beneficial in particular circumstances. Sexual reproduction offers several important advantages. First, offspring have a mixture of genes from both parents, which produces variation in the population. This variation increases the chance of a species surviving environmental changes. If conditions shift, some individuals may have characteristics that help them adapt and survive. For example, if the environment changes and introduces new predators, individuals with slightly different characteristics might be better suited to escape these predators. They're more likely to survive and reproduce, passing their beneficial genes to the next generation. This principle forms the basis for natural selection. Individuals with advantageous characteristics are more likely to survive, reproduce, and pass their genes to the next generation. Over time, this leads to better adaptation of the species to its environment and can result in evolutionary change. Humans have harnessed this process through selective breeding to develop animals and plants with desirable characteristics. For example, we've bred animals that produce more meat or milk and plants that yield larger fruits or are more resistant to disease. However, sexual reproduction also has drawbacks. It requires finding a mate, which takes time and energy. The search for a partner can also expose animals to predators. Additionally, only 50% of an individual's genes are passed to each offspring, meaning that favorable combinations of genes might be lost in the next generation. Asexual reproduction offers different advantages. Since only one parent is needed, organisms can reproduce without finding a mate, saving considerable energy. This method is typically faster than sexual reproduction, allowing organisms to produce many offspring quickly when conditions are favorable. The speed of asexual reproduction makes it particularly useful for colonizing new environments rapidly. When a single bacterial cell reaches a new location with plenty of nutrients, it can quickly multiply to form a large population. Another advantage is that 100% of the parent's genes are passed to the offspring. If the parent is well adapted to its environment, all offspring will inherit these adaptations. However, the lack of genetic variation in asexually reproducing populations can be a disadvantage. If environmental conditions change, the entire population might be susceptible to the same threats, such as new diseases or environmental stresses. Interestingly, some organisms can reproduce both sexually and asexually, depending on their circumstances. The malaria parasite provides a fascinating example. When a mosquito carrying the parasite bites a human, the parasite reproduces asexually in the human host. However, when it returns to a mosquito, it reproduces sexually. Many species of fungi reproduce both sexually and asexually. They produce spores asexually, which can become new fungi when they land in a suitable place. But they can also reproduce sexually, introducing variation into their populations. Sexually produced spores introduce variation and are often produced in response to unfavorable environmental changes. This increases the chance that the population will survive the change. Plants often demonstrate both strategies as well. Many flowering plants produce seeds through sexual reproduction, but can also reproduce asexually. Strawberry plants send out runners, stems that grow horizontally along the ground. New plants form at intervals along these runners, genetically identical to the parent. Daffodils and other bulb plants can form new bulbs from the main bulb. Each new bulb can grow into a genetically identical plant. Asexual reproduction in plants can take place in different ways. Some plants produce runners. These are stems that grow horizontally on the surface of the soil away from the parent plant. At various points along the runner, a new strawberry plant forms that is identical to the parent plant. Other examples include plants that grow from bulbs, like daffodils. New bulbs can form from the main bulb and divide off. Each new bulb can grow into a new identical plant. The ability to use both reproductive strategies gives these organisms flexibility. They can reproduce sexually to generate genetic diversity, helping the species adapt to changing conditions. In our next episode, we'll delve deeper into meiosis, the special type of cell division that produces gametes with half the normal number of chromosomes. In this episode, we'll look at why gametes need half the usual number of chromosomes. Next, we'll examine the process of meiosis and how it differs from mitosis. Then we'll explore how meiosis creates genetic variation. Finally, we'll see what happens after fertilization when gametes combine. In sexual reproduction, offspring receive genetic information from two parents. As we discussed in our previous episode, when two gametes join during fertilization, they form a cell with the full complement of chromosomes. For this to work properly, gametes must contain half the normal chromosome number. Otherwise, the fertilized cell would have too many chromosomes, which would disrupt normal development. Normal body cells in humans contain 46 chromosomes, 23 from each parent. To maintain this number across generations, gametes must have just 23 chromosomes. Meiosis is the process that produces these specialized cells with half the chromosome count. It occurs in the reproductive organs, the testes in males and the ovaries in females. Unlike mitosis, which produces two identical cells, meiosis involves two sequential cell divisions, resulting in four genetically different cells. Let's look at how this works. Before meiosis begins, the cell duplicates its genetic information. Each chromosome makes an exact copy of itself, forming two identical strands attached at a central point. In the first division of meiosis, something unique happens. The duplicated chromosomes arrange themselves in pairs in the center of the cell. These pairs consist of matching chromosomes, one originally from the mother and one from the father. They line up along the middle of the cell. The pairs are then pulled apart, with one copy of each chromosome moving to opposite ends of the cell. After this first division, we have two cells, each containing half the number of chromosome pairs, but each chromosome still consists of two attached strands. In the second division of meiosis, these cells divide again. The chromosomes line up in the center once more, and the two strands of each chromosome are pulled apart. This results in four cells, each containing just one set of chromosomes, half the number found in normal body cells. These are our gametes. An important feature of meiosis is that it creates genetic diversity. During the process, genetic material from the mother's and father's chromosomes can exchange in a process called crossing over. Additionally, the way chromosomes separate during meiosis is random. This shuffling of genetic material means that each gamete has a unique combination of genes. This is why siblings can look quite different from each other. They inherit different combinations of genes from their parents. After fertilization, when two gametes fuse, the resulting cell contains the normal number of chromosomes again, half from each parent. This cell, called a zygote, then divides by mitosis to produce lots of new cells as an embryo develops. As the embryo grows, these cells differentiate to form the various specialized cell types that make up a complete organism. Meiosis is crucial for sexual reproduction and genetic diversity. Without it, the chromosome number would double with each generation, which would be incompatible with life. And without the genetic reshuffling that occurs during meiosis, offspring would not have the genetic variation that helps species adapt and evolve. In our next episode, we'll explore DNA and the genome, the genetic instructions that determine the characteristics of living organisms. In this episode, we'll explore DNA and the genome, the remarkable molecules that contain the genetic instructions for all living organisms. We'll examine the structure of DNA, take a look at genes, explore protein synthesis, and finally, we'll discuss mutations. Understanding genetics starts with getting to grips with DNA and genes. DNA stands for deoxyribonucleic acid and is the chemical that all of the genetic material in a cell is made up from. DNA contains genetic information, basically all the instructions to put an organism together and make it work. It's what's in your DNA that determines what inherited characteristics you have. DNA is found in the nucleus of animal and plant cells in really long structures called chromosomes. Chromosomes normally come in pairs. In humans, we have 23 pairs, making a total of 46 chromosomes. DNA is made up of two strands coiled together in the shape of a double helix. This twisted ladder structure is critical to how DNA functions. Let's look more closely at what DNA is made of. DNA strands are polymers made up of lots of repeating units called nucleotides. Each nucleotide consists of three parts: a sugar, a phosphate group, and a base. The sugar and phosphate groups in the nucleotides form the backbone of the DNA strands. The sugar and phosphate groups alternate, with the phosphate groups linking the sugars together. One of four different bases, A, T, C, or G, joins to each sugar. These letters stand for adenine, thymine, cytosine, and and the two strands of DNA are held together by bonds between the bases. Importantly, the bases don't pair up randomly. A always pairs up with T, and C always pairs up with G. This is called complementary base pairing. This complementary nature of DNA is crucial for both DNA replication and protein synthesis. It ensures that genetic information can be copied accurately. A gene is a small section of DNA found on a chromosome. Each gene codes for a particular sequence of amino acids, which are put together to make a specific protein. Only 20 amino acids are used, but they make up thousands of different proteins. Genes simply tell cells in what order to put the amino acids together. Each amino acid is coded for by a sequence of three bases in the gene. This three-base sequence is often called a codon. The amino acids are joined together to make various proteins depending on the order of the gene's bases. This in turn determines what type of cell it is, like a red blood cell. It's the order of bases in a gene that decides the order of amino acids in a protein. Let's turn our attention to how proteins are made using the information in DNA. This process is called protein synthesis. Proteins are made in the cell cytoplasm on tiny structures called ribosomes. To make proteins, ribosomes use the code in the DNA. But there's a problem. DNA is found in the cell nucleus and can't move out of it because it's really big. So the cell needs to get the code from the DNA to the ribosomes. This is done using a molecule called mRNA, which is made by copying the code from the nucleus. The mRNA acts as a messenger between the genetic code in the nucleus and the ribosomes. It carries the message between the two. The correct amino acids are brought to the ribosomes in the correct order by carrier molecules. Once the chain of amino acids has been assembled, it folds into a unique shape. This folding is crucial. It gives the protein its specific three-dimensional structure, which allows it to perform its particular function. The shape of a protein is directly related to its role in the body. Proteins have many different functions in the body. Let's look at some examples. Enzymes act as biological catalysts to speed up chemical reactions in the body. For instance, digestive enzymes help break down food. Hormones, like insulin, are used to carry messages around the body. Insulin is released into the blood by the pancreas to regulate blood sugar levels. Structural proteins, like collagen, are physically strong. They strengthen connective tissue, such as ligaments and cartilage. Every organism has a genome. The genome is just a fancy term for the entire set of genetic material in an organism. Scientists have worked out the complete human genome. Understanding the human genome is a really important tool for science and medicine for many reasons. It allows scientists to identify genes in the genome that are linked to different types of disease. Knowing which genes are linked to inherited diseases could help us understand them better and develop effective treatments for them. Scientists can also look at genomes to trace the migration of certain populations of people around the world. All modern humans are descended from a common ancestor in Africa, but humans can now be found all over the planet. The human genome is mostly identical in all individuals, but as different populations of people have split off in different directions, they gradually developed small differences in their genomes. By investigating these differences, scientists can work out when new populations split off and what paths they took. Sometimes the sequence of DNA bases can be changed. These changes are called mutations. Mutations are random changes in an organism's DNA, and they can sometimes be inherited. Mutations occur continuously. They can occur spontaneously, such as when a chromosome isn't replicated properly. However, the chance of mutations is increased by exposure to certain substances or some types of radiation. Mutations change the sequence of the DNA bases in a gene, which produces a genetic variant, a different form of the gene. As the sequence of DNA bases codes for the sequence of amino acids in a protein, mutations to a gene can sometimes lead to changes in the protein that it codes for. Most mutations have little or no effect on the protein. Some will change it in such a small way that its function or appearance is unaffected. However, some mutations can seriously affect a protein. Sometimes the mutation will code for an altered protein with a change in its shape. This could affect its ability to perform its function. For example, if the shape of an enzyme's active site is changed, the substrate may no longer be able to fit into it. Structural proteins like collagen could lose their strength if their shape is changed, making them pretty useless at providing structure and support. If there's a mutation in the non-coding DNA, it can alter how genes are expressed. There are different types of mutations. Insertions are where a new base is inserted into the DNA base sequence where it shouldn't be. You should remember that three bases in a DNA base sequence codes for a particular amino acid. An insertion changes the way the groups of three bases are read, which can change the amino acids that they code for. Insertions can change more than one amino acid as they have a knock-on effect on the bases further on in the sequence. Deletions are when a random base is deleted from the DNA base sequence. Like insertions, they change the way that the base sequences are read and can have knock-on effects further down the sequence. Substitutions happen when a random base in the DNA base sequence is changed to a different base. A substitution may change the amino acid that is coded for, potentially affecting the protein structure and function. Understanding DNA, genes, the genome, and mutations helps us appreciate the remarkable complexity of life at the molecular level. These tiny structures and processes determine who we are and how our bodies function. They represent the basic code of life itself, instructions passed down through countless generations. In our next episode, we'll explore how these genetic principles apply to genetic inheritance. In this episode, we'll explore genetic inheritance. Key topics we'll discuss today include how genes control characteristics, different forms of genes called alleles, genetic diagrams, and family trees. Many genes can directly control what characteristics you develop. Different genes control different characteristics. Some characteristics are controlled by a single gene, like mouse fur color and red-green color blindness in humans. However, most characteristics are controlled by several genes interacting together. This makes inheritance patterns more complex for traits like height and skin color. All genes exist in different versions called alleles. These are represented by letters in genetic diagrams, one letter for each allele. You have two versions or alleles of every gene in your body, one from each parent. These alleles can be the same or different from each other. If an organism has two alleles for a particular gene that are the same, then it's homozygous for that trait. If the alleles for a particular gene are different, then it's heterozygous. Sometimes one allele can completely override the other when they're different. The allele that shows itself is called the dominant allele. We typically use a capital letter for dominant alleles. For example, capital A. The other one is called the recessive allele, represented by a small letter, for example, lowercase a. A recessive allele only shows itself when there's no dominant allele present. For an organism to display a recessive characteristic, both its alleles must be recessive. To display a dominant characteristic, the organism only needs one dominant allele. This means the dominant allele overrides the recessive one if the organism is heterozygous. Your genotype is the combination of alleles you have, while your phenotype are the observable characteristics you have. Let's see how we can predict inheritance patterns using genetic diagrams, also known as Punnett squares. Suppose we're breeding chickens with long legs. The allele which causes chickens to have long legs is recessive and represented by a lowercase h, while normal legs are due to a dominant allele, represented here with a capital H. Let's look at what happens if you breed from two heterozygous chickens. We start by identifying the parents' genotypes. Both parents are heterozygous. Next, we determine what types of sex cells each parent can produce. We can then create a Punnett square to show all possible combinations. We place one parent's potential gametes along the top and the other parent's along the side. Filling in the square gives us the following. Looking at the phenotypes, we can see that 3 of the offspring will have normal legs, while 1 will have long legs. This gives us a 3-to-1 ratio of normal to long-legged offspring in this generation. Now, if 2 of these offspring breed together, what happens in the next generation? It depends on which offspring breed together. If a homozygous dominant breeds with a heterozygous chicken, all their offspring will be normal. On the other hand, if a heterozygous chicken breeds with a homozygous recessive chicken, half their offspring will be normal and half will have long legs. Let's look at another example. A lion with long hair, which is the dominant trait, is bred with a lion with short hair, which relates to the recessive trait. The long-haired lion has a heterozygous genotype, while the short-haired lion has a homozygous recessive genotype. When we work out the Punnett square, we find the offspring will have a 1-to-1 ratio, half with long hair and half with short hair. This happens because the heterozygous parent can pass either the dominant or recessive allele to its offspring. The homozygous recessive parent can only pass the recessive allele. Understanding genetic inheritance can help us interpret family trees, which is particularly useful for tracking genetic disorders like cystic fibrosis. From a family tree, we can often tell if an allele for a particular disorder is dominant or recessive. For cystic fibrosis, we know it's a recessive disorder because plenty of people carry the allele but don't have the disease. By analyzing a family tree, we can also determine the probability that someone will inherit a genetic disorder. For example, if both parents are carriers for a recessive disorder but don't show symptoms, there's a 25% chance their child will have the disorder. Similarly, there's a 50% chance their child will be a carrier like them, and a 25% chance the child will neither have the disorder nor be a carrier. The risk calculation for the new baby is just the same as in the genetic diagram, so a new baby could be unaffected, be a carrier, or have cystic fibrosis. Genetic diagrams are important tools in understanding and predicting inheritance patterns. They allow us to calculate the probability of particular traits appearing in offspring based on the parents' genotypes. These diagrams help us understand why certain traits might skip generations or appear unexpectedly. For example, if two brown-eyed parents have a blue-eyed child, genetic diagrams explain that both parents must carry a recessive allele for blue eyes. It's important to remember that genetic diagrams show probabilities. They don't guarantee exactly what will happen with each individual offspring. Just like flipping a coin doesn't guarantee exactly 50% heads and 50% tails, in a small number of flips, genetic inheritance can sometimes produce unexpected ratios in small family sizes. In reality, many traits are influenced by multiple genes and environmental factors, making inheritance patterns more complex than the simple examples we've discussed. However, these principles form the foundation for understanding how genes are passed from one generation to the next. In our next episode, we'll explore how these inheritance patterns can lead to genetic disorders when certain mutations are passed down through families. In this episode, we'll explore inherited disorders. We'll start by outlining what inherited disorders are, then discuss examples such as cystic fibrosis and polydactyly, and finally look at embryo screening and the ethical questions it raises. Some disorders can be inherited from your parents through the genes they pass on to you. These genetic conditions are caused by mutations in specific genes that lead to health problems. Let's start by looking at cystic fibrosis, which is a genetic disorder affecting cell membranes. It results in the body producing a lot of thick, sticky mucus in the air passages and in the pancreas. Cystic fibrosis is caused by a recessive allele. This means that for a person to have the condition, they must inherit two copies of the recessive allele, one from each parent. Because it's recessive, people with only one copy of the allele won't have the disorder. They're known as carriers. For a child to have cystic fibrosis, both parents must be either carriers or have the disorder themselves. As genetic diagrams show, if both parents are carriers, there's a 1 in 4 chance that their child will have the condition. Now let's look at polydactyly, which is quite different in terms of how it's inherited. Polydactyly is a genetic disorder where a baby is born with extra fingers or toes. It doesn't usually cause any other problems, so it's not life-threatening. Unlike cystic fibrosis, polydactyly is caused by a dominant allele. This means the disorder can be inherited if just one parent carries the defective allele. As the genetic diagram shows, there's a 50% chance of a child having the disorder if one parent has it. Modern medicine now allows for screening of embryos for genetic disorders. This process is done during in vitro fertilization. In IVF, embryos are fertilized in a laboratory and then implanted into the mother's womb. Before being implanted, it's possible to remove a cell from each embryo and analyze its genes. Many genetic disorders can be detected in this way, such as cystic fibrosis. It's also possible to get DNA from an embryo in the womb and test it for disorders. There are lots of ethical, social, and economic concerns surrounding embryo screening. Embryonic screening is quite controversial because of the decisions it can lead to. For embryos produced by IVF after screening, embryos with bad alleles would be destroyed. For embryos in the womb, screening could lead to the decision to terminate the pregnancy. There are strong arguments both for and against embryonic screening. Those against embryonic screening worry that it implies people with genetic problems are undesirable. This could increase prejudice. They also express concerns about where to draw the line. Could parents eventually select for traits like eye color or intelligence? Others note that screening is expensive and question whether it's a good use of resources. On the other hand, supporters of embryonic screening argue that it will help to stop people suffering. They point out that treating genetic disorders costs the government and taxpayers a lot of money. Some also believe there are limits to how far screening should go. But at the moment, parents cannot even select the sex of their baby unless it relates to a health reason. Understanding inherited disorders and the technologies available to detect them raises important questions about the intersection of science, medicine, and ethics. While advances in genetic screening offer new possibilities for preventing suffering, they also challenge us to consider difficult questions about what kinds of interventions are appropriate and where the boundaries should be. In our next episode, we'll explore sex determination and how the combination of chromosomes determines whether a baby will be male or female. In this brief episode, we'll explore how your biological sex is determined at the moment of conception by specific chromosomes. Understanding this process helps explain one of the fundamental aspects of human development. Let's begin by recalling what we've learned about chromosomes. There are 23 pairs of chromosomes in every human body cell. Of these, 22 pairs are matched pairs called autosomes that control various characteristics. The 23rd pair is special. They're the sex chromosomes labeled X or Y. These two chromosomes decide your sex, whether you develop as male or female. Males have an X and a Y chromosome, giving them the combination XY. It's the Y chromosome that causes male characteristics to develop. Without it, male features wouldn't appear. Females have two X chromosomes, giving them the combination XX. This XX combination allows female characteristics to develop. During gamete formation, something important happens. When making sperm cells, the X and Y chromosomes are separated during meiosis, the special cell division we discussed in a previous episode. This separation means that each sperm cell gets either an X chromosome or a Y chromosome, never both. There's a 50% chance a sperm cell will contain an X chromosome and a 50% chance it will contain a Y chromosome. A similar process happens when females produce X cells. However, since females only have X chromosomes, all X cells will contain an X chromosome. When fertilization occurs, the sex of the future child depends on which sperm cell fertilizes the egg. If a sperm cell carrying an X chromosome fertilizes the egg, the resulting combination will be XX, a female. If a sperm cell carrying a Y chromosome fertilizes the egg, the resulting combination will be XY, a male. We can use genetic diagrams to illustrate this process by creating a simple Punnett square with the mother's possible gametes along one side and the father's possible gametes. Along the top, we can see all possible combinations. The resulting square shows two XX outcomes and two XY outcomes, demonstrating that there's an equal probability, 50-50, of having a boy or girl in each pregnancy. This 50-50 ratio is only a probability in each individual pregnancy. Having a boy doesn't make it more likely the next child will be a girl. Each pregnancy is an independent event with the same 50% probability. Understanding sex determination helps us appreciate how fundamental biological characteristics are established at conception through the combination of chromosomes. No two living things are exactly alike. In this episode, we'll explore variation, the differences that make each organism unique. We'll look at how genes contribute to differences in organisms, how the environment influences characteristics, and how mutations introduce new variation. You've probably noticed that all people are different from one another. These differences aren't just limited to humans. All species show variation. Different species obviously look different. My dog definitely doesn't look like a dairy cow. But even organisms of the same species will usually look at least slightly different. In a room full of people, you'll see different eye colors, differently shaped noses, and various hair colors. These differences are called variation within a species, and there are two main types: genetic variation and environmental variation. Genetic variation comes from our genes. All plants and animals have characteristics that are similar in some ways to their parents. This happens because an organism's characteristics are determined by the genes they inherit. Genes are the codes inside your cells that control how you're made. These genes are passed on in sex cells or gametes, from which the offspring develop. Most animals and quite a lot of plants get some genes from the mother and some from the father. The combining of genes from two parents causes genetic variation. No two of the species are genetically identical except for identical twins. Some characteristics are determined only by genes. In humans, these include eye color, blood group, and certain inherited disorders like hemophilia or cystic fibrosis. However, characteristics are also influenced by the environment. The environment, including the conditions that organisms live and grow in, causes differences between members of the same species. For example, a plant grown in plenty of sunlight would be green, but the same plant grown in darkness would be tall and spindly with yellow leaves. These are environmental variations. Environmental variation covers a wide range of differences, from losing a finger in an accident to getting a suntan to plants having yellow leaves. Most characteristics are actually due to a mixture of genes and the environment. For example, your maximum height that you could possibly grow to is determined by your genes, but whether you actually reach that height depends on environmental factors like nutrition. Mutations also introduce variation. Mutations are changes to the sequence of bases in DNA. As we learned earlier, mutations can lead to changes in the protein that a gene codes for. Most mutations have no effect on the protein and therefore don't alter characteristics significantly. Although it's very rare, mutations can result in a new phenotype being seen in a species. If the environment changes and the new phenotype makes an individual more suited to the new environment. It can become common throughout the species relatively quickly by natural selection. In our next episode, we'll explore natural selection and how it drives evolution. The theory of evolution explains how all species on Earth today descended from simple life forms that first appeared over 3 billion years ago. We'll cover Darwin's revolutionary insights into natural selection, the process of speciation, evidence supporting evolutionary theory, and finally, extinction factors. Let's start with Darwin's groundbreaking observations and conclusions. Charles Darwin developed his theory based on careful observations made during his 5-year voyage aboard HMS Beagle. Darwin recognized two crucial facts about living organisms that became foundational to his theory. First, individuals within any species show lots of variation in their characteristics, from size and color to behavior and physiology. Second, organisms constantly compete for limited resources in their environment. From these observations, Darwin reasoned that individuals with characteristics better suited to their environment would succeed more in this competition. These better adapted organisms would survive longer, find more food, avoid predators more effectively, and ultimately produce more offspring. This principle became known as survival of the fittest, though fittest means best adapted to the environment, not necessarily strongest. The organisms with advantageous traits would pass these beneficial characteristics to their offspring through inheritance. Conversely, organisms with less favorable characteristics would struggle to survive and reproduce. Their genes would gradually become less common in the population, potentially disappearing entirely. Over many generations, beneficial characteristics become increasingly prevalent whilst disadvantageous ones diminish. This gradual change in populations over time is evolution, driven by the mechanism of natural selection. Evolution explains both the unity and diversity of life on Earth. All species share common ancestors if we trace back far enough, explaining fundamental similarities like cellular structure and DNA. The differences arise because populations have adapted to vastly different environments over millions of years. Darwin's ideas have been refined with modern scientific discoveries, particularly in genetics. We now understand that genes control inheritance, and variation arises from mutations, random changes in DNA sequences. When beneficial mutations occur, natural selection favors these variants, potentially spreading them throughout the population. This genetic understanding, unavailable to Darwin, has strongly validated his fundamental insights about evolution's mechanisms. Over extended periods, accumulated changes can become so significant that populations can no longer interbreed successfully. This process, called speciation, creates entirely new species from existing ones. For speciation to occur, it typically requires reproductive isolation between populations. Reproductive isolation often occurs when populations become geographically separated by barriers like mountain ranges, rivers, or ocean expanses. These separated populations face different environmental pressures and food sources, driving divergent evolutionary paths. As isolated populations adapt to their specific environments, they accumulate different genetic changes. Eventually, they become so genetically distinct that they cannot produce fertile offspring together, even if reunited. Once reproductive isolation is complete, the populations represent separate species following independent evolutionary trajectories. This process has generated Earth's millions of species from common ancestors over billions of years. Multiple lines of evidence support evolution by natural selection, though Darwin initially faced skepticism. When On the Origin of Species was published in 1859, the supporting evidence was less comprehensive than today. The fossil record provides compelling evidence for evolution. Fossils preserve remains of ancient organisms in rock layers, with older fossils in deeper layers. This record shows countless extinct species, demonstrating that Earth's inhabitants have changed dramatically over time. Fossils reveal gradual transitions between ancient and modern forms. The evolution of horses, for example, can be traced from small, multi-toed ancestors to modern single-hoof species through numerous intermediate fossils. Comparing anatomy, which means structure, gives us another line of evidence through homologous or similar structures. The front limbs of humans, cats, whales, and bats contain the same basic bone arrangements despite serving different functions. These structural similarities suggest descent from a common ancestor. Vestigial structures provide further anatomical evidence for evolution. The human appendix, wisdom teeth, and tailbone serve little or no function but make sense as remnants from ancestors who used them. Some snakes retain tiny, internal leg bones despite having no legs. These vestigial structures would be inexplicable without evolution but perfectly logical as inheritance from legged ancestors. Modern molecular biology has provided powerful new evidence through DNA and protein comparisons. Species sharing recent common ancestors have more genetic similarities than those whose lineages deviated earlier. Humans and chimpanzees share approximately 98% of their DNA, reflecting our recent evolutionary divergence. In contrast, humans share less genetic similarity with more distantly related species. We can observe evolution occurring in real time with rapidly reproducing organisms. Bacterial evolution of antibiotic resistance demonstrates natural selection within human timescales. When exposed to antibiotics, susceptible bacteria die, whilst resistant variants survive and multiply, creating resistant populations. Darwin's theory initially faced opposition for several reasons beyond scientific skepticism. It contradicted prevailing religious beliefs about divine creation, offering a naturalistic explanation for life's diversity. Darwin couldn't fully explain how traits were inherited or how new variations arose. The science of genetics, which would later support his theory, remained undiscovered for decades after his publication. Limited fossil evidence at the time made Darwin's claims difficult to verify conclusively. Alternative evolutionary theories competed with Darwin's natural selection. Jean-Baptiste Lamarck proposed that organisms could pass acquired characteristics to offspring, suggesting that traits developed during life would be inherited. Lamarck's theory predicted that a giraffe stretching to reach high leaves would develop a longer neck, passing this trait to offspring. Similarly, a blacksmith's children would inherit his muscular arms developed through work. However, experiments consistently disproved Lamarckian inheritance. We now know that acquired physical changes don't alter genetic information. If you lose a finger in an accident, your children aren't born with missing fingers. The discovery of genetics validated Darwin's theory by explaining inheritance mechanisms. Mendel's work on heredity, though initially overlooked, eventually merged with evolutionary theory to form modern evolutionary synthesis. Additional fossil discoveries have revealed many transitional forms between major groups. Archaeopteryx, discovered shortly after Darwin published his theory, showed features of both reptiles and birds. Tiktaalik, found in 2004, demonstrates the transition from fish to tetrapods with both fins and primitive limb bones. Extinction represents evolution's other phase, the disappearance of species unable to adapt sufficiently. The fossil record reveals that over 99% of all species that ever lived are now extinct. Species become extinct when environmental changes exceed their ability to adapt. For example, climate shifts, habitat destruction, or new predators can eliminate populations before beneficial mutations arise and spread. New diseases can devastate species lacking immunity. Competition from better adapted species can drive others to extinction through resource depletion. Invasive species often outcompete native organisms, leading to local or global extinctions. Catastrophic events cause mass extinctions, eliminating numerous species simultaneously. The asteroid impact 66 million years ago killed the dinosaurs along with approximately 75% of all species. Earth has experienced five major mass extinctions, each followed by evolutionary radiations as survivors diversified. These events have profoundly shaped life's history, creating opportunities for new groups to dominate. Human activities are currently driving what scientists call the sixth mass extinction. Habitat destruction, pollution, overharvesting, and climate change are eliminating species faster than natural background rates. Current extinction rates exceed normal levels by 100 to 1,000 times. Understanding evolution helps us appreciate life's interconnectedness and predict species' responses to environmental changes. This knowledge proves crucial for conservation efforts and managing human impacts on biodiversity. Evolution by natural selection, supported by evidence from anatomy, molecular biology, and direct observation, stands as one of science's most robust theories. It explains life's diversity whilst revealing the common threads connecting all organisms. Next time, we'll explore how humans apply evolutionary principles through selective breeding of plants and animals. Today, we'll explore selective breeding, a technique humans have used for thousands of years to develop plants and animals with characteristics we find useful or attractive. In this episode, we'll take a look at what selective breeding actually is, the steps involved in this process, examples of selective breeding in action, and issues associated with selective breeding. Selective breeding might sound like a complicated topic, but it's actually really straightforward. It's when humans choose plants or animals with characteristics they want to keep and make sure these organisms reproduce together. This way, the genes for those useful or attractive features remain in the population. Think about it. We've developed cows that produce tons of milk, crops that resist plant diseases, and dogs with friendly personalities. None of these existed naturally. Humans created them through selective breeding. The process is super simple. First, from your existing stock of plants or animals, you pick out the ones with the characteristics you're looking for. Then you breed these selected organisms with each other. From their offspring, you again select the ones showing the best examples of the trait you want and breed them together. You keep this up for several generations, and the desirable trait gets stronger and stronger. Eventually, all the offspring have the characteristic you wanted. Farmers use selective breeding all the time to improve yields. For example, to get more milk, a farmer might breed together the cows and bulls with the best milk producing traits. After doing this for several generations, they end up with cows that produce way more milk than their ancestors ever did. To get more meat, they might select cattle with genes for larger size. Selective breeding isn't some new technology. People have been doing it for thousands of years. It's how we ended up with edible crops from wild plants, and how we got domesticated animals like cows and dogs. Before modern science, farmers and animal breeders didn't know about genes, but they still observed which individuals had the best traits and bred them together. But selective breeding isn't perfect. It has some serious drawbacks. The main problem is that it reduces what scientists call the gene pool, the number of different alleles or forms of a gene in a population. We covered alleles in our episode on genetic inheritance. This happens because the breeder keeps breeding from similar animals or plants which are all closely related. This is known as inbreeding. Inbreeding can cause health problems because there's more chance of harmful recessive defects when the gene pool is limited. Some dog breeds are particularly susceptible to certain defects because of inbreeding, like bulldogs having breathing problems. There can also be serious issues if a new disease appears. Because there's not much variation in the population and all the stock are closely related to each other, if one of them is going to be killed by a new disease, the others are likely to succumb to it too. This lack of genetic diversity means if environmental conditions change, these selectively bred organisms might not have the genetic variation needed to adapt and survive. Understanding selective breeding helps us appreciate how humans have shaped the domesticated plants and animals we rely on today. It also highlights the importance of maintaining genetic diversity in our food sources. In our next episode, we'll explore genetic engineering, a more recent technology that allows scientists to modify the genetic material of organisms directly. Genetic engineering represents one of the most powerful technologies in modern biology, enabling scientists to transfer genes between completely unrelated organisms. In this video, we'll look at what genetic engineering is, how scientists transfer genes between organisms, real-world applications, and benefits and concerns of genetic engineering. Let's start by defining genetic engineering and its unique capabilities. Genetic engineering involves extracting genes responsible for desirable characteristics from one organism and inserting them into another organism's genome. This process transfers specific traits directly between species that could never naturally interbreed. Unlike selective breeding, which requires many generations of controlled reproduction to enhance existing traits, genetic engineering achieves precise modifications immediately. Scientists can add characteristics from bacteria to plants or from humans to bacteria, combinations impossible through conventional breeding. The genetic engineering process follows a systematic sequence of carefully controlled steps. First, scientists identify and isolate the specific gene responsible for the desired characteristic using restriction enzymes. Restriction enzymes function as molecular scissors, cutting DNA at precise locations to extract the target gene. These enzymes recognize specific DNA sequences, ensuring accurate removal of the desired genetic material. The isolated gene must then be inserted into a vector, a biological vehicle for gene delivery. Common vectors include bacterial plasmids, which are small circular DNA molecules, or modified viruses that can penetrate cells efficiently. Different organisms require different vectors for successful gene transfer. Plant cells might receive genes through bacterial vectors, whilst animal cells often require viral vectors designed to integrate with mammalian DNA. The vector carries the gene into target cells like a molecular delivery system. Once inside target cells, the new gene integrates into the organism's genome, becoming part of its genetic blueprint. If modification occurs early in development, such as in embryos, the entire organism develops with a new characteristic. Successful integration means the organism's cells produce proteins coded by the inserted gene. These proteins create the desired characteristics, whether insulin production in bacteria or pest resistance in crops. Genetic engineering has produced remarkable medical breakthroughs, particularly in pharmaceutical production. Bacteria modified with human insulin genes now produce vast quantities of this essential hormone for diabetics. This bacterial insulin is identical to human insulin, eliminating previous reliance on animal sources. Before genetic engineering, diabetics depended on insulin extracted from pig pancreases, which sometimes caused allergic reactions. Now, genetically modified bacteria produce pure human insulin safely and efficiently in controlled fermentation tanks. Scientists have engineered bacteria to manufacture numerous therapeutic proteins, including human growth hormone for treating dwarfism and clotting factors for hemophilia patients. These medicines would be impossible to produce affordably without genetic modification. Agricultural applications have created crops with enhanced characteristics benefiting both farmers and consumers. Genetically modified plants can resist diseases, tolerate herbicides, and repel insect pests without chemical pesticides. Some corn varieties produce bacterial proteins toxic to caterpillars but harmless to humans. This built-in pest resistance reduces crop losses and decreases pesticide use, potentially benefiting the environment. Gene therapy represents genetic engineering's most ambitious medical application. Researchers are developing treatments that insert functional genes into patients with genetic disorders. This approach could potentially cure conditions like cystic fibrosis by replacing faulty genes with working versions. Despite these benefits, genetic engineering generates significant controversy and legitimate concerns. Critics worry about unforeseen long term consequences that might only emerge after widespread adoption. Environmental concerns focus on genetically modified crops potentially affecting ecosystems. Modified plants might crossbreed with wild relatives, spreading engineered traits unpredictably through natural populations. Reduced biodiversity represents another ecological concern. GM crops might harm beneficial insects or reduce wildflower populations on farmland. These changes could cascade through food webs, affecting birds and other wildlife dependent on agricultural ecosystems. Consumer concerns about GM foods persist despite extensive safety testing showing no increased health risks. Many people prefer avoiding genetically modified products, driving demand for labeling and non GM alternatives. Ethical questions arise about humanity's right to alter genetic codes fundamentally. Should we modify organisms for our benefit? And where should we draw boundaries on this powerful technology? Next time, we'll explore cloning, another biotechnology raising scientific and ethical questions. In this episode, we'll cover plant cloning methods, animal cloning techniques, Dolly the sheep, and the challenges and concerns associated with cloning. Let's start with the world of plant cloning. Plants can be cloned through tissue culture, a laboratory technique using small samples of plant cells. These cells are placed in growth medium containing specific nutrients and hormones that stimulate development into complete plants. Tissue culture offers several advantages over traditional propagation methods. Plants can be produced rapidly in minimal space, grown throughout the year regardless of season, and multiplied from tiny amounts of parent material. One of the benefits of cloning is that scientists can use tissue culture to preserve endangered plant species that struggle to reproduce naturally. Commercial plant breeders employ tissue culture to mass produce desirable varieties quickly. A single exceptional plant can generate thousands of identical offspring within months, ensuring consistent quality for agriculture and horticulture. Taking cuttings represents a much older cloning method that gardeners have practiced for centuries. This involves removing a portion of a healthy parent plant, typically a stem or leaf, and encouraging it to develop roots. Cuttings provide a quick, inexpensive way to produce genetically identical plants. Many common garden plants, from roses to tomatoes, can be successfully propagated this way without specialized equipment. Both methods produce clones, organisms genetically identical to their parent. This ensures desirable traits like disease resistance, fruit quality, or flower color are preserved exactly in offspring. Animal cloning requires far more complex procedures than plant cloning. Embryo transplants allow farmers to produce multiple offspring from their highest quality livestock. This technique begins with collecting sperm from a prize bull and eggs from an elite cow. The eggs are fertilized artificially in laboratory conditions, creating embryos with superior genetics. Each embryo is then split multiple times whilst cells remain unspecialized, creating numerous identical embryos. These cloned embryos are implanted into surrogate mothers who carry them to term. The resulting calves are genetically identical to each other, though not to the surrogate mothers who birthed them. This technique can produce hundreds of elite offspring annually from a single genetic pairing. Adult cell cloning represents a more revolutionary approach, creating copies from mature organisms. This process begins by removing the nucleus from an unfertilized egg cell, essentially creating an empty cellular vessel. Scientists then extract the nucleus from an adult body cell, such as a skin or udder cell. This nucleus, containing the complete genetic information, is inserted into the enucleated egg cell. An electric shock stimulates the reconstructed egg, triggering cell division as if fertilization had occurred. Once the embryo develops into a ball of cells, it's implanted into a surrogate mother's womb. This technique famously produced Dolly the sheep in 1996. Dolly demonstrated that specialized adult cells could be manipulated to create entire organisms. Her birth revolutionized our understanding of cellular development and opened new possibilities for biotechnology. Despite technological advances, cloning presents significant challenges and concerns. The primary issue involves reduced genetic diversity in cloned populations. Genetic uniformity makes populations vulnerable to diseases and environmental changes. If all individuals share identical genes, a single pathogen could potentially eliminate entire populations lacking resistance. Cloned animals often experience serious health problems. Dolly developed arthritis at an unusually young age, raising questions about premature aging in clones. Many cloned animals suffer organ dysfunction, immune system abnormalities, and shortened lifespans. The success rate for animal cloning remains extremely low, with numerous failures for each success. These failures raise ethical concerns about animal welfare and the justification for continuing such research. However, cloning research offers potential benefits, including preserving endangered species and advancing medical understanding. Studying cloned animals provides insights into genetic disorders, aging processes, and developmental biology. Next time, we'll explore speciation, examining how species change over time. In this episode of our series, we'll look at what speciation actually means, population isolation, the role of natural selection in all of this, and the person behind the theory, Alfred Russel Wallace. Evolution, as we learned in a previous episode, leads to the development of lots of different species over time. This process of creating new species is called speciation. Let's take a look at what this means. A species is a group of similar organisms that can reproduce to give fertile offspring, while speciation is the development of a new species. Speciation occurs when populations of the same species become so different that they can no longer successfully interbreed to produce fertile offspring. In other words, they've evolved into separate species. How does this happen? It starts with isolation, where populations of a species become separated, often due to a physical barrier appearing between them. For example, floods and earthquakes can create barriers that geographically isolate some individuals from the main population. Mountains rising up, rivers changing course, or even roads in cities can separate populations. Imagine a population of beetles living in a forest that gets split by a new motorway being built right through the middle. The beetles on one side can no longer mate with the beetles on the other side because they simply can't cross the busy road. This physical separation is the first crucial step towards speciation. When populations are isolated, the conditions on either side of the barrier will be slightly different. They might have different climates, different predators, or different food sources. This may mean different characteristics will become more common in each population due to natural selection operating differently. Let's continue with our beetle example to see how this works. Perhaps one side of the motorway has darker soil, whilst the other side has lighter, sandy ground. On the dark soil side, beetles with darker coloration will be better camouflaged from predators like birds. Meanwhile, on the sandy side, lighter colored beetles will have the advantage because they blend in better with their surroundings. Each population shows genetic variation because they have a wide range of alleles. In each environment, individuals with characteristics that make them better adapted to their surroundings have a better chance of survival and are more likely to breed successfully. So the alleles that control these beneficial characteristics are more likely to be passed on to the next generation. Over time, the gene pool of each population changes in different ways. Think of it like two separate breeding programs happening simultaneously, each selecting for different traits. After many generations, our beetle populations might become so genetically different that even if the motorway was removed, they could no longer produce fertile offspring together. The dark beetles and light beetles would have become entirely separate species through the process of speciation. An important scientist in developing the theory of speciation was Alfred Russel Wallace. He was working at the same time as Charles Darwin and was one of the early scientists exploring the idea of speciation. Wallace's contributions to evolutionary theory were absolutely groundbreaking, yet he often gets overshadowed by Darwin's fame. During his career, Wallace independently came up with the idea of natural selection and published work on the subject together with Darwin in 1858. This prompted Darwin to publish On the Origin of Species the following year. Wallace made observations as he traveled around the world that provided lots of evidence to support the theory of evolution by natural selection. For example, he is most famous for his work on warning colors on insects which were used by some species to deter predators from eating them. Wallace's research took him to remote places like the Amazon rainforest and the Malay archipelago, where he collected thousands of specimens. His detailed observations of how species varied between different islands helped build the foundation of our modern understanding of speciation. Understanding speciation helps us see how the incredible diversity of life on Earth has developed. It's the process that has created the millions of different species we see today, and it continues to happen all around us. In our next episode, we'll explore the fascinating evidence for evolution from the fossil record. This episode of our series covers the work of Gregor Mendel on genetics, why his ideas weren't popular at the time, and further developments in genetics after him. Let's begin with Gregor Mendel, an Austrian monk whose work in the 1860s laid the foundation for modern genetics. He carried out a series of breeding experiments using pea plants, selecting them because they had clear, contrasting features. What made Mendel's approach special was that he carefully tracked specific traits such as plant height, flower color, and seed shape through multiple generations. Think about it. He literally counted thousands of individual pea plants and precisely recorded their characteristics. Through these painstaking experiments, Mendel observed that traits weren't simply blended together in offspring as was commonly believed at the time. Instead, traits appeared to be passed down as discrete units that remained unchanged from generation to generation. Mendel called these units factors, what we now know as genes. He concluded that each trait was determined by two factors, one inherited from each parent, and that some factors were dominant over others. For example, when he crossed tall pea plants with short ones, the offspring weren't medium height as people expected. Instead, they were all tall, showing that the tall factor was dominant over the short factor. Despite publishing his findings in 1866, Mendel's work remained largely unrecognized during his lifetime. But why was such groundbreaking research ignored? There were several reasons for this scientific oversight. First, Mendel published his work in an obscure scientific journal that wasn't widely read by the international scientific community. Second, his statistical approach to biology was unusual for the time. Most biologists weren't mathematically trained and struggled to understand his numerical analysis. The idea of using maths to study living things was quite revolutionary. Perhaps most importantly, the scientific community simply wasn't ready for Mendel's ideas. His theories contradicted the prevailing belief that inherited traits were blended together, like mixing paint colors. There was also no understanding of the physical basis for his mysterious factors. Scientists couldn't see or touch them. It wasn't until the early 20th century that the significance of his work was finally recognized. The rediscovery of Mendel's research coincided perfectly with new observations about cell biology. In the late 19th century, scientists had developed much better microscopes that allowed them to observe chromosomes during cell division. They could see these thread-like structures moving around inside cells, but didn't yet understand what they did. This was like having a key but not knowing what door it unlocked. In the early 20th century, scientists began to notice something remarkable. Chromosomes and Mendel's factors behaved in remarkably similar ways during reproduction. Both seemed to be passed from parent to offspring in predictable patterns. This led to the crucial insight that genes might actually be located on chromosomes. This connection between Mendel's abstract factors and the physical structures inside cells was a major breakthrough in scientific understanding. Scientists now had a mechanism to explain how true traits could be inherited. The chromosomes were like tiny filing cabinets carrying genetic information. But the story doesn't end there. In the mid 20th century, another huge advance came with the discovery of DNA structure by Watson and Crick in 1953. This built on the work of many other scientists, including Rosalind Franklin, whose X-ray crystallography was crucial to understanding DNA's shape. Once the double helix structure of DNA was understood, scientists could begin to work out exactly how genes function at the molecular level. They discovered that genes contain the instructions for making proteins, which carry out most of the important functions in our cells. All of this scientific work by many different scientists over more than a century led to the development of what we now call gene theory. This is our modern understanding of how characteristics are inherited through genes and how they function in our bodies. This story of the development of genetic understanding is a perfect example of how science progresses. Ideas are proposed, tested, sometimes forgotten, then rediscovered and refined as technology and knowledge advance. Scientific understanding rarely comes from a single breakthrough, but develops gradually through the collective work of many scientists over time. Each discovery builds upon previous work, creating an ever growing tower of knowledge. Our next episode will explore the evidence for evolution, including fossils and antibiotic resistance in bacteria. This episode will cover what fossils actually are, the different ways they form, how they show organisms change over time, and why there are gaps in our understanding. So, what exactly are fossils? Fossils are the preserved remains or traces of organisms that lived long ago. They're typically found embedded in rock and can be millions of years old. Fossils are incredibly valuable to scientists because they provide evidence of what ancient creatures looked like and how they've evolved over time. They help us piece together the history of life on Earth. There are three main ways that fossils can form. Let's take a look at each of these processes. The first and most common way fossils form is through gradual replacement of minerals. This happens when parts of an organism that don't easily decay, such as bones, shells, and teeth, are buried in sediment. Over very long periods, these hard parts are gradually replaced by minerals as they decay. The minerals form a rock-like substance that takes the shape of the original body part. Eventually, the surrounding sediments turn to rock, with the fossil clearly visible within it. When someone discovers and carefully excavates the rock, the fossil is revealed. The second way fossils can form is through casts and impressions. Sometimes, an organism may leave an imprint in soft material like mud or clay. Later, material hardens around the imprinted area, leaving behind a cast. Things like footprints, burrows, or plant roots can also create impressions in soft sediments, which are preserved when the material hardens. These trace fossils don't contain any actual parts of the organism, but they provide evidence of its existence and behavior. The third way fossils form is through preservation in environments where decay doesn't occur. In certain special places, organisms can be preserved almost intact. For example, in amber, fossilized tree resin, insects and small creatures can be trapped and preserved with remarkable detail. The resin prevents oxygen and decay microbes from reaching the organism. Similarly, in glaciers, the extreme cold stops decay microbes from working. In peat bogs, the highly acidic conditions prevent decay organisms from breaking down tissue. A remarkable example of this type of preservation was the discovery of woolly mammoths in Siberian permafrost, with some specimens so well preserved that their skin, hair, and even internal organs remained intact after thousands of years. Fossils give us crucial information about how living things have changed as life has developed on Earth over millions of years. By comparing fossils from different time periods, scientists can track how species have evolved. However, despite all we've learned from fossils, there's still a major mystery. How did life begin on Earth? There are various scientific hypotheses about how the first living things appeared, but there's no complete consensus. Some suggest that the first life emerged in primordial oceans or near deep sea vents. Perhaps simple organic molecules came together on early Earth, combined to form more complex organic molecules, and eventually developed into very simple life forms. These hypotheses are difficult to confirm because we lack solid evidence from that time period. Many early forms of life were soft bodied, with tissues that easily decayed. This means the fossil record for the earliest life forms is incomplete. Additionally, many fossils that did form millions of years ago may have been destroyed by geological activity, such as the movement of tectonic plates. The fossil record is a bit like a book with missing pages. Scientists can read the parts that remain and try to piece together the complete story, but there will always be gaps in knowledge. Despite these limitations, the study of fossils has revolutionized our understanding of life's history on Earth. Each new fossil discovery adds another piece to this grand puzzle, helping us better understand the amazing story of life on our planet. In this episode of our series, we'll explore when antibiotic medicine started, how bacteria began outsmarting antibiotics, antibiotic resistance, and finally, how the world is responding to this issue. Let's begin with one of medicine's greatest success stories. The discovery of antibiotics like penicillin was revolutionary. Before antibiotics, a simple cut could kill you if it got infected. Imagine living with that constant fear. Suddenly, bacterial infections that had been death sentences became curable with pills. But here's where things get interesting. Bacteria have developed an incredible survival trick. They can evolve to resist the very drugs designed to destroy them. How on earth does this happen? Like all living things, bacteria sometimes get random mutations in their DNA. Think of it as nature rolling the dice. Most mutations do nothing, but occasionally one hits the jackpot. Some lucky bacteria might develop the ability to break down antibiotics like they're digesting food. Others evolve tiny pumps that literally push the antibiotic out of their cells before it can do damage. This is how bacteria develop resistance against medicines. And once they have these traits, they pass them on to their offspring. How wonderful. When you take antibiotics, the non resistant bacteria die off quickly, but those resistant ones survive, thrive, and have the whole environment to themselves. With all their competition dead, resistant bacteria multiply like crazy. They can produce millions of copies of themselves in just hours. We're talking about new generations every 20 minutes. Plus, they can share genetic information with each other in the form of plasmids. This means resistance can spread through bacterial populations at lightning speed. One resistant bacterium can quickly become millions, all carrying the same resistance genes. For people infected with these superbugs, standard treatments become less helpful. Take MRSA, for example. This notorious superbug can be incredibly difficult to treat. If MRSA enters your bloodstream, it can be fatal. For decades, we've been winning the war against bacterial infections. Death rates from diseases like pneumonia have plummeted thanks to antibiotics. But now we're losing ground, and it's partly our own fault. Doctors sometimes prescribe antibiotics for minor illnesses or viral infections like colds and flu. Here's the problem: antibiotics can't kill viruses. It's like trying to use a hammer on a screw. Additionally, patients make things worse by not finishing prescriptions. They feel better after a few days and stop taking the pills, but this leaves the strongest bacteria alive. Every time we use antibiotics incorrectly, we're essentially training bacteria to become resistant. We're creating the perfect conditions for superbugs to evolve. Remember, antibiotics don't create resistance directly. They simply kill off the weak bacteria, leaving the playground clear for resistant ones to take over. That's why finishing your full course of antibiotics is crucial. It ensures all bacteria are destroyed, not just the weak ones. The problem extends beyond hospitals and clinics. On farms, antibiotics are sometimes given to healthy animals to make them grow faster and prevent illness. This creates a breeding ground for resistant bacteria. These superbugs can then jump to humans through meat production and consumption. Some countries have started restricting agricultural antibiotic use, recognizing the danger. But globally, we still use massive amounts of antibiotics in farming. Pharmaceutical companies are racing to develop new antibiotics that can defeat resistant strains. But here's the catch: creating new drugs is painfully slow and expensive. This is why prevention is vital. Using antibiotics only when truly necessary, completing full treatments, and limiting agricultural use can all slow resistance development. What we're witnessing is evolution in action, natural selection at hyperspeed. Bacteria are adapting to survive in a world filled with antibiotics, just as Darwin predicted organisms would adapt to environmental changes. Next time, we'll explore how scientists organize and classify the incredible diversity of life on Earth. In this episode of our series, we'll look at why classification is important, the Linnaean system, how molecular biology led to new systems, and evolutionary trees. Let's explore the principles behind biological classification. Classification is fundamental to how humans process information, from organizing libraries to structuring databases. In biology, this organizational instinct becomes a vital scientific tool. By grouping the sheer number of organisms on Earth based on shared characteristics, scientists can identify patterns and understand relationships between different forms of life. Without classification, biological research would lack the structure needed for meaningful analysis. The modern classification system originated with Carl Linnaeus, an 18th century Swedish scientist. He developed the Linnaean system, which arranges organisms into nested groups, each level more specific than the last. This hierarchical structure creates a logical framework for understanding biological diversity. The classification hierarchy consists of 7 main levels: kingdom, phylum, class, order, family, genus, and species. Each level represents a different degree of relatedness between organisms. This system functions like a postal address, beginning with broad categories and progressively narrowing down. Just as an address moves from country to specific street number, classification moves from kingdom to individual species. For over 2 centuries, this framework provided the foundation for biological classification. Traditionally, scientists recognized 5 kingdoms: animals, plants, fungi, protists, and bacteria. This division was based primarily on visible characteristics and basic cellular structure. However, advances in microscopy and molecular biology revealed that traditional classification based on physical appearance didn't accurately reflect evolutionary relationships. Scientists discovered that organisms appearing similar externally could be very different at the molecular level. In 1990, Carl Woese proposed a revolutionary three-domain system based on genetic analysis, which transformed our understanding of life's most basic organization. Through RNA sequence analysis made possible by new technology, Woese identified 3 fundamental divisions of life that supersede the kingdom level. The 3 domains are archaea, bacteria, and eukaryota. These represent the earliest evolutionary branches in the tree of life. Archaea are single-celled prokaryotes, often found in extreme environments, such as thermal vents and salt lakes. Despite superficial similarities to bacteria, they possess unique biochemical characteristics. These organisms demonstrate remarkable adaptations to conditions lethal to most other life forms. The bacteria domain encompasses true bacteria, including familiar species like E. coli and Staphylococcus. Though prokaryotic like archaea, bacteria have distinct molecular and biochemical properties. Eukaryotes include all organisms with nucleated cells and membrane-bound organelles. This domain contains the kingdoms of fungi, plants, animals, and protists. This classification reveals surprising relationships. Humans share more fundamental characteristics with fungi than bacteria share with archaea. Such insights demonstrate the value of molecular classification. Within these three domains, the traditional Linnaean hierarchy remains useful. The system continues to provide detailed classification below the domain level. Scientific naming follows strict international conventions to ensure clarity. The binomial nomenclature system assigns each species a two-part Latin name. The first part indicates the genus, while the second identifies the specific species. For example, humans are designated Homo sapiens, Homo denoting our genus, and sapiens our species. This systematic approach eliminates vagueness in scientific communication. Binomial nomenclature provides a universal language for scientists worldwide. Regardless of native language or location, researchers use identical names for the same organisms. Evolutionary trees visually represent relationships between species based on common ancestry. These tools help scientists understand how different organisms evolved from shared ancestors. Branch points on these trees indicate common ancestors, with closer branches suggesting more recent divergence. The structure reveals evolutionary relationships that may not be apparent from physical characteristics alone. Scientists construct these trees using multiple data sources, including DNA sequences, protein structures, and fossil evidence. Modern computational methods allow analysis of vast amounts of genetic data. This comprehensive approach provides insights into evolutionary history spanning billions of years. By integrating various evidence types, scientists can trace relationships from microorganisms to complex multicellular life. Next time, we'll examine biodiversity and explore why maintaining species variety is critical for ecosystem stability. This episode will look at key ecological terminology, how organisms compete for limited resources, the concept of interdependence, and finally, how changes to one species can cascade through an entire community. Let's start by defining the fundamental concepts that ecologists use to understand the natural world. Ecology is the scientific study of organisms, their environment, and the complex interactions between them. First, let's understand the precise terminology ecologists use to talk correctly about environmental issues. A habitat is the physical location where an organism lives and obtains all its requirements for survival and reproduction. Examples include tropical forests, coral reefs, Arctic tundra, freshwater lakes, and even the human gut where bacteria thrive. Each habitat provides specific conditions that determine which organisms can successfully survive and reproduce there. A population consists of all individuals of one species living within a particular habitat at a given time. For instance, all fish of a particular species in the sea. A community comprises all the different populations that interact within a habitat, forming complex networks of relationships. These species interact through feeding relationships, competition for resources, cooperation, and various other ecological connections that bind them together. Environmental factors are classified as either abiotic or biotic, a distinction that helps scientists understand different influences on organisms. Abiotic factors include non-living components such as temperature, light intensity, water availability, and many more factors. Biotic factors encompass all living components within an environment, including plants, animals, fungi, bacteria, and other microorganisms. These organisms constantly interact through predation, competition, mutualism, parasitism, and numerous other ecological relationships. An ecosystem combines the community with its abiotic environment, forming a functional unit where energy flows and nutrients cycle. Within ecosystems, organisms require specific resources for survival, growth, and reproduction. Plants need light for photosynthesis, space for their body to grow, water for metabolic processes, and mineral ions from soil for building proteins and other essential molecules. Animals require territory for living, hunting, and breeding, food for energy and growth, water for survival, and mates for reproduction. The availability of these resources directly influences population sizes, distribution patterns, and the overall structure of ecological communities. Since resources are finite in any environment, organisms must compete for them continuously. This competition for resources for survival occurs both within species, also known as intraspecific competition, and between different species, which is known as interspecific competition. In forest ecosystems, trees compete intensely for sunlight, extending their branches and leaves to capture maximum light energy. This competition creates a layered structure of forests with tall trees above, shade-tolerant shrubs in the middle, and specially adapted plants growing on the dark forest floor. Species better adapted to compete for specific resources will dominate their environment, whilst less competitive species may be forced to occupy different ecological niches, migrate elsewhere, or face local extinction. This influences population sizes in habitats. Beyond competition, interdependence exists between species for various services essential to survival, creating an intricate web of dependencies that maintains ecosystem stability through carefully balanced relationships developed over thousands of years of evolution. Plants depend on insects for pollination and animals for seed dispersal, whilst providing food, shelter, and oxygen in return. Decomposers like bacteria and fungi break down dead material, recycling nutrients that plants require for growth, completing the circle of life. These interconnections mean that changes to one species can trigger cascading effects throughout the entire community. Consider a stream ecosystem containing algae, larvae, and fish. Larvae serve as primary consumers, feeding on algae and controlling its growth whilst preventing harmful algal blooms. They also provide food for predators like fish, making them a crucial link in the aquatic food web. Without larvae consuming algae, algal populations may proliferate excessively, potentially creating blooms that reduce oxygen levels and affect water quality. This example illustrates how removing a single species disrupts the entire ecosystem balance profoundly. Each change triggers further adjustments as the community attempts to reach a new equilibrium, which may be very different from the original stable state. Some communities achieve remarkable stability despite constant small fluctuations in population sizes. Stable communities maintain relatively constant population sizes through balanced predator prey relationships, cooperation between species, and balance with environmental factors. These systems demonstrate remarkable resilience to minor disturbances whilst maintaining their overall structure and function over time. By studying these complex interactions, we gain insights into maintaining biodiversity and ecosystem health. Next time, we'll examine living and non living factors in more detail. This episode of the series covers the differences between abiotic and biotic factors, how these factors affect different organisms and populations, and how they combine to create complex ecosystems. Let's start by examining the non living components that shape life on Earth. Abiotic factors are the non living, physical, and chemical components of an ecosystem that determine which organisms can exist in particular environments. Key abiotic factors include moisture levels in soil and air, which influence water availability for all organisms. Light intensity determines photosynthesis rates in plants and affects animal behavior patterns. Temperature influences metabolic rates, enzyme function, and activity levels across all living things. Carbon dioxide levels are crucial for plant photosynthesis, whilst oxygen levels determine survival for aquatic animals. Wind intensity affects seed dispersal, pollination, and physical stress on organisms. Finally, soil pH and mineral content, particularly nitrate deficiency, directly influence plant growth due to inefficient protein synthesis. These factors determine which plant species can thrive, subsequently affecting entire food webs built upon plant producers. Changes in abiotic factors can trigger profound ecosystem responses. Consider what happens when light intensity decreases due to prolonged cloud cover or canopy closure. Reduced light intensity limits photosynthesis rates in plants, decreasing their energy production. This leads to slower growth rates, reduced reproduction, and potentially declining plant populations. The effects cascade through the ecosystem as herbivores lose food sources and shelter. Predator populations subsequently decline as their prey becomes scarce, demonstrating the interconnected nature of ecosystem responses. Biotic factors represent the living components and their interactions within ecosystems. These factors include all the ways organisms influence each other's population and community sizes. Predation is a fundamental biotic factor, with predators controlling prey populations whilst depending on them for survival. The introduction of new predators can dramatically alter community dynamics, sometimes leading to population crashes or extinctions. Competition occurs when organisms require the same limited resources. This competition can be intraspecific between members of the same species or interspecific between different species occupying similar ecological niches. Consider red and gray squirrels inhabiting the same woodland. Both species compete for similar food sources and nesting sites, with the better adapted species typically dominating over time. Pathogens represent another crucial biotic factor, causing diseases that can decimate populations. Historical data often shows dramatic population crashes following pathogen introduction. A population increasing steadily might suddenly plummet when disease strikes, taking years or decades to recover to previous levels. Food availability directly controls population sizes, as organisms cannot survive without adequate nutrition. Changes in food supply, whether through environmental changes or species interactions, immediately affect dependent populations. The interaction between living and non living factors creates particularly complex ecosystem dynamics. Temperature changes might favor certain pathogens, whilst rainfall patterns influence plant growth and subsequent herbivore populations. These interactions make ecosystem responses difficult to predict, but fascinating to study. By understanding how abiotic and biotic factors interact, ecologists develop conservation strategies to protect vulnerable ecosystems. This scientific understanding guides management decisions for maintaining biodiversity and ecosystem services essential for human well being. This episode in our series explores what adaptations are, their three main types, specific examples in animals, and extremophiles. Let's start by understanding what adaptations are and how they develop. Adaptations are inherited characteristics that increase an organism's chances of survival and reproduction in its environment. These features develop over many generations through natural selection, where beneficial traits become more common in populations. Scientists classify adaptations into three main types, each serving different survival functions. Structural adaptations involve physical features of an organism's body, including shape, size, and coloration. These anatomical characteristics directly influence how organisms interact with their environment. Arctic foxes exemplify structural adaptation through their seasonal color changes. Their white winter fur provides essential camouflage against snow, helping them avoid predators whilst hunting prey effectively. In summer, their coat turns brown, matching the tundra landscape. Temperature regulation requires different structural adaptations depending on climate. Cold adapted animals like whales possess thick blubber layers and low surface area to volume ratios, minimizing heat loss. Conversely, desert animals such as camels have thin fat layers and high surface area to volume ratios. These features maximize heat dissipation, preventing overheating in extreme temperatures. Behavioral adaptations involve actions organisms take to enhance survival. Migration represents a crucial behavioral adaptation for many species. Swallows travel thousands of kilometers seasonally, following food availability and suitable breeding conditions. These behavioral patterns can be instinctive or learned, including feeding strategies, predator avoidance, and social interactions. Each behavior increases survival probability in specific environmental conditions. Functional adaptations are internal physiological processes that enhance survival. These include metabolic adjustments, reproductive strategies, and biochemical mechanisms that help organisms cope with environmental challenges. Desert animals demonstrate remarkable functional adaptations for water conservation. They produce minimal sweat and highly concentrated urine, reducing water loss dramatically. These physiological adjustments allow survival in environments where water is extremely scarce. Hibernation represents another functional adaptation, particularly in brown bears. During winter, they dramatically reduce their metabolic rate, conserving energy when food is unavailable. This metabolic slowdown allows bears to survive months without eating, relying on stored body fat. Heart rate and breathing decrease significantly, reducing energy requirements to minimal levels. Microorganisms display the most extreme adaptations, with some species thriving in conditions lethal to most life forms. These extremophiles have revolutionized our understanding of life's limits. Some bacteria flourish in volcanic vents exceeding 100 degrees Celsius, temperatures that would denature most proteins. Other extremophiles inhabit hypersaline lakes with salt concentrations several times higher than seawater. They've evolved special mechanisms to prevent cellular dehydration in these extreme conditions. Deep sea bacteria survive crushing pressures that would destroy most organisms. Their adapted proteins and cellular structures function normally under pressures thousands of times greater than at sea level. Studying adaptations provides crucial insights into evolutionary processes and biodiversity. Each adaptation represents millions of years of evolutionary refinement, perfectly matching organisms to their ecological niches. This knowledge becomes increasingly important for conservation efforts and ecosystem management. By understanding how organisms adapt, we can better protect vulnerable species and maintain ecological balance. In this episode, we'll begin by examining food chains. Next, we'll trace energy movement through feeding relationships. And finally, we'll take a look at predator prey relationships. Let's begin with the fundamental concept of food chains. A food chain is a linear sequence that shows how energy and nutrients pass within an ecosystem from one organism to another through feeding. All food chains begin with organisms capable of producing their own food, known as producers. These organisms form the essential foundation of all life on Earth by creating biomass from inorganic substances. Producers, typically plants or algae, manufacture glucose through the process of photosynthesis. This remarkable biochemical process converts light energy from the sun into chemical energy stored in glucose molecules. When plants produce glucose, some is used immediately for respiration to release energy for cellular processes. The remainder gets converted into other biological molecules such as starch, cellulose, and proteins that build the plant's structure. These molecules collectively form what scientists term biomass, the total mass of living material in an organism. Biomass represents stored chemical energy that originated from sunlight captured during photosynthesis. This energy transfers through the ecosystem when one organism consumes another, though the transfer is never completely efficient. At each stage, substantial energy is lost through respiration, movement, and heat production. Primary consumers are herbivores that feed directly on producers. These herbivores, such as rabbits, deer, or caterpillars, obtain their energy by consuming plant material. They convert plant biomass into animal biomass through the processes of digestion. Primary consumers subsequently become food for secondary consumers, which are carnivores that feed on herbivores. Secondary consumers may then be consumed by tertiary consumers, creating additional levels in the food chain. The numbers of organisms at each level demonstrate the inefficiency of energy transfer. Consider this simplified example of numbers. Thousands of plants might support hundreds of caterpillars, which in turn could sustain tens of small birds, and that may support one eagle. We must now examine the dynamic relationship between predators and prey. Predators are animals that capture and consume other animals, whilst prey are the animals being consumed. In ecosystems containing both predator and prey species, population dynamics follow predictable patterns. These populations don't remain constant, but instead fluctuate in regular cycles that maintain ecosystem balance. Initially, prey populations are typically limited by the availability of their food resources. When conditions are favorable and food is abundant, prey populations can increase substantially. As prey numbers increase, predators have access to more food, which improves their survival and reproduction rates. Consequently, the predator population begins to grow in response to this increased food availability. However, as predator numbers rise, they consume increasing quantities of prey. This increased predation pressure causes the prey population to decline significantly. With fewer prey available, predators face food scarcity, which leads to reduced predator survival and reproduction, causing their population to decrease. As predator numbers fall, predation pressure on prey diminishes, allowing prey populations to recover. This recovery of prey numbers eventually provides more food for predators, initiating another cycle. The process continues indefinitely, creating fluctuations in both populations. Significantly, these population cycles are not synchronized but show a characteristic time lag. Changes in prey population are followed by corresponding changes in predator population after a delay. This delay occurs because population responses require time to manifest. When prey numbers decline, predators don't immediately disappear as they can survive temporarily on reduced food or stored energy reserves. Understanding food chains and predator prey relationships enables ecologists to predict ecosystem responses to environmental changes. This knowledge proves essential for effective conservation strategies and ecosystem management. In our next episode, we'll examine how materials like carbon and water cycle through ecosystems, returning to the environment for reuse by living organisms. In this episode in our series, we'll examine how materials cycle through ecosystems. Then we'll dive into the water cycle. Next, we'll take a look at the carbon cycle. And finally, how these cycles create interdependence between all living things. All materials in the living world undergo constant recycling through natural processes, ensuring the continuation of life on Earth. Without these cycles, essential materials would become permanently locked away and unavailable for use by living things. Every living organism requires specific materials to survive, grow, and reproduce. Plants need water, carbon dioxide, and mineral nutrients, whilst animals require water, oxygen, and organic nutrients obtained from their food. The source of these materials lies in Earth's remarkable natural cycles. Let's examine the water cycle, which represents the continuous movement of water throughout our planet. The water present on Earth today has been recycled countless times over geological history. The total quantity of water on Earth remains essentially constant over time. However, this water continuously changes form and location through various physical and biological processes. The water cycle begins when solar energy causes water to evaporate from oceans, lakes, rivers, and soil surfaces. This process transforms liquid water into water vapor, which enters the atmosphere. Plants contribute to this process through transpiration, where water absorbed by roots moves through the plant and exits through stomata in leaves. Together, evaporation and transpiration add substantial quantities of water vapor to the atmosphere. This warm water vapor rises through the atmosphere due to its lower density. As water vapor ascends to higher altitudes, it encounters cooler temperatures, causing it to condense into droplets. These droplets aggregate to form clouds, which can transport water over considerable distances. Eventually, when water droplets within clouds become sufficiently large and heavy, they fall as precipitation. This precipitation may take the form of rain, snow, or hail, depending on atmospheric conditions. This precipitation provides the fresh water essential for terrestrial life. Without this continuous supply, land-based organisms could not survive, as ocean water contains salt concentrations toxic to most terrestrial species. Once precipitation reaches land, it follows various pathways back to the ocean. Some water infiltrates the soil to become groundwater, where plant roots can absorb it for photosynthesis and other metabolic processes. Other water flows across the surface as runoff, forming streams and rivers that eventually drain back into seas and oceans. This completes the cycle, allowing the process to begin again. The water cycle is critical because only approximately 3% of Earth's water exists as fresh water. Much of this fresh water remains frozen in ice caps and glaciers, making it unavailable for immediate use. The continuous cycling ensures that the limited liquid freshwater remains accessible for living organisms. Now we must examine another fundamental cycle, the carbon cycle. Carbon atoms form the backbone of all organic molecules, including carbohydrates, proteins, lipids, and nucleic acids. Plants absorb elements like carbon, oxygen, hydrogen, and nitrogen from their environment. They convert these simple inorganic substances into complex organic compounds that constitute their biomass. These materials pass through food chains as organisms consume one another. Eventually, all materials return to the environment through excretion, death, and decomposition. The carbon cycle is particularly significant for maintaining life on Earth. Carbon dioxide from the atmosphere is absorbed by plants and algae during photosynthesis. This carbon becomes incorporated into glucose molecules, which can be converted into various organic compounds that form plant structures. When plants and algae respire, some carbon returns to the atmosphere as carbon dioxide. This represents only a portion of the carbon they initially absorbed, as much remains in their biomass. When animals consume plants, carbon compounds transfer through the food chain from primary consumers to secondary and tertiary consumers. Each organism uses some carbon for respiration, releasing carbon dioxide back to the atmosphere. When organisms die, decomposers and detritus feeders break down their remains. These microorganisms respire as they feed, releasing carbon dioxide and completing this portion of the cycle. Animals also produce waste products containing carbon compounds. Decomposers break down this waste material, returning carbon to the atmosphere through their respiration. Human activities, particularly burning fossil fuels, have significantly increased atmospheric carbon dioxide levels. This represents an acceleration of carbon release that would otherwise occur over geological timescales. In stable ecosystems, materials removed from the soil by plants are balanced by those returned through decomposition. This equilibrium maintains nutrient availability and sustains ecosystem productivity over time. These cycles, though conceptually straightforward, are absolutely vital for life's continuation on Earth. Without them, essential materials would become depleted, causing ecosystem collapse. This episode in our series will cover what decomposition is, how it produces compost, environmental factors affecting rate of decomposition, and how this process can provide a renewable source of energy. Decomposition is essential for maintaining the flow of nutrients through ecosystems. Microorganisms break down plant and animal material to obtain energy for metabolism, energy that would otherwise remain locked in dead organic matter. The process of decay occurs continuously in natural environments worldwide. For example, in forests, fallen leaves decompose on the forest floor, and in gardens, organic waste breaks down in compost heaps. Compost is decomposed organic matter that can be used as a natural fertilizer for plants. It contains essential nutrients released during the decomposition process, making it valuable for improving soil fertility and structure. Farmers and gardeners manipulate environmental conditions to accelerate decomposition and produce high quality compost efficiently. The primary agents of decomposition are microorganisms, including bacteria and fungi, which secrete enzymes to break down complex organic molecules. Larger organisms called detritus feeders, such as woodlice and earthworms, also contribute by physically breaking down material into smaller pieces. These decomposers work together to convert dead organic matter into simpler substances. The smaller particles created by detritus feeders provide increased surface area for microorganisms to colonize and digest. Several environmental factors significantly influence the rate of decomposition. Understanding these factors allows us to predict decomposition rates in different environments and manipulate conditions for practical applications. Temperature is the first major factor affecting decomposition rate. Warmer temperatures generally accelerate decomposition because heat increases enzyme activity and weakens cellular structures in dead material. However, extremely high temperatures can denature enzymes essential for decomposition, whilst very cold temperatures dramatically slow the process. The optimal temperature range for most decomposers lies between 25 and 35 degrees Celsius. Outside this range, decomposition rates decline significantly, which explains why organic matter decomposes slowly in Arctic environments but rapidly in tropical regions. Oxygen availability represents another crucial factor in decomposition. Most decomposer organisms require oxygen for aerobic respiration, which provides the energy needed for breaking down organic matter. In oxygen-rich environments, aerobic decomposition proceeds rapidly and completely, converting organic matter into carbon dioxide, water, and mineral nutrients. When oxygen is limited, different microorganisms carry out anaerobic decomposition, which proceeds more slowly and produces different end products. Water availability also plays a critical role in decomposition processes. Decomposer organisms require water for their metabolic processes, including enzyme production and nutrient transport. Moisture also helps dissolve organic compounds, making them more accessible to microorganisms. Decomposition occurs most rapidly in moist environments, typically when water content is between 50 and 60% of the material's water holding capacity. Both excessive moisture, which can limit oxygen availability, and drought conditions significantly reduce decomposition rates. The abundance of decomposer organisms directly affects how quickly organic matter breaks down. Areas with diverse and numerous decomposer populations experience faster decomposition than those with limited decomposer communities. By manipulating these factors, humans can control decomposition rates for various purposes. Gardeners maintain optimal conditions in compost heaps by turning them regularly to provide oxygen, maintaining moisture levels, and sometimes adding materials to adjust temperature. Now we must examine biogas production through anaerobic decay. Biogas consists primarily of methane, which can be burned as a fuel for heating, cooking, or electricity generation. It's produced when organic material decomposes in the absence of oxygen through a process called anaerobic decay. Specialized anaerobic microorganisms break down plant and animal waste without oxygen, producing methane as byproduct. This natural process occurs in waterlogged soils, landfill sites, and the digestive systems of ruminant animals. Biogas production facilities at sewage treatment works and agricultural sites harness this process on an industrial scale. These facilities use specially designed containers called digesters or generators to optimize conditions for anaerobic decomposition. These digesters must maintain constant temperatures, typically around 35 degrees Celsius, to keep anaerobic microorganisms functioning efficiently. Temperature fluctuations can severely reduce methane production or halt the process entirely. Unlike natural gas, biogas cannot be stored as a liquid under pressure and must be used immediately. The immediate use requirement means biogas generators are typically connected directly to heating systems, cooking facilities, or electricity generators. This limitation affects how biogas systems are designed and implemented. Two main types of biogas generators exist: batch generators, and continuous generators. Each type has specific advantages depending on the scale and consistency of organic waste availability. Batch generators process waste in discrete batches, with material loaded, left to digest completely, and then cleared before the next batch. These systems suit situations with periodic waste availability but require more labor to operate. Continuous generators receive constant waste input and produce biogas at steady rates. These systems work well for large-scale operations with consistent waste streams, such as sewage treatment plants or large farms. Understanding decomposition reveals the remarkable efficiency of natural recycling systems. Without decomposers, dead organic matter would accumulate indefinitely, and essential nutrients would become unavailable to living organisms. The continuous activity of decomposers maintains the flow of materials through ecosystems supporting all life on Earth. Humans have learned to manipulate decomposition processes for practical benefits, producing nutrient-rich compost for agriculture and renewable energy through biogas generation. These applications demonstrate how understanding natural processes can provide sustainable solutions to human needs. We'll cover what environmental change means for living organisms, analyze three key environmental factors that determine species distribution, and consider the various causes of environmental change. Environmental changes alter the geographical ranges where species can maintain viable populations to live and reproduce successfully. These changes affect organisms differentially, causing some species to expand their ranges whilst others decline. Understanding these responses requires exploring specific environmental factors that limit species distribution. Water availability represents the first main factor influencing species distribution. All organisms require water for metabolic processes, though requirements vary considerably between species. Water availability often determines the boundaries of species' geographical ranges, particularly in dry regions. In tropical regions, species distribution shift predictably between wet and dry seasons. These seasonal rainfall differences create temporary changes in habitat suitability that determine distributional changes. The East African wildebeest migration demonstrates this phenomenon. Millions of individuals migrate northward once the dry season starts in the south. They follow rainfall patterns and resulting grass growth in the north, then return south when rains resume there. Temperature represents the second critical factor determining species distributions. Even modest temperature changes can substantially impact where species survive by altering habitat thermal suitability. Many species have narrow temperature tolerances that restrict them to specific climatic zones. Current climate warming causes observable distributional shifts worldwide. Rising average temperatures in Germany have enabled Mediterranean species to expand northward into previously unsuitable regions. The European bee-eater, historically restricted to Mediterranean areas, now breeds regularly in parts of Germany. This northward expansion directly corresponds to warming temperatures, making previously cold regions thermally suitable. Atmospheric composition constitutes the third important distributional factor. Different species exhibit varying sensitivities to carbon dioxide concentrations and air pollutants. Certain species demonstrate extreme sensitivity to air quality, making them valuable environmental bioindicators. Lichen species exemplify organisms highly sensitive to pollution levels. Different lichens exhibit varying pollution tolerances, with some thriving only in pristine air, whilst others tolerate moderate pollution. The absence of pollution-sensitive lichens from urban areas demonstrates how atmospheric composition controls distribution. Similarly, industrial processes affecting water chemistry cause sensitive fish species to disappear from polluted waters. Environmental changes occur through various mechanisms at different scales. Some changes represent natural variations to which species have adapted over evolutionary time. Seasonal variations create predictable changes that species exploit through migration, dormancy, or reproductive timing. Geographic differences across latitudes and altitudes create stable environmental gradients determining species distributions. These natural patterns have shaped species adaptations over millennia. However, human activities increasingly drive rapid environmental changes exceeding natural variation rates. Rising global temperatures, primarily from human-caused greenhouse gas emissions, force species to shift ranges towards the poles of Earth and upward in elevation. Many species cannot disperse quickly enough to track rapidly changing conditions. Others face dispersal barriers like urban development, preventing range shifts. These constraints lead to population declines and local extinctions when species cannot escape deteriorating conditions. Understanding how environmental changes affect species distributions helps scientists predict future biodiversity patterns and design effective conservation strategies. Efforts focus on maintaining habitat connectivity through protected areas and wildlife corridors, enabling species to move and adapt to shifting conditions. Monitoring species distributions provides early warning signals of environmental change impacts. By understanding how environmental factors control distributions, we can better predict and manage human impacts on biodiversity. In this episode, we'll cover what biodiversity is, how population growth increases environmental pressure through resource consumption, the growing global waste problem, and then consider strategies for protecting Earth's biological diversity. Biodiversity refers to the variety of different species of organisms on Earth or within particular ecosystems. This encompasses all forms of life, including plants, animals, fungi, and microorganisms. It also covers the genetic variation, which contributes to the complex web of interactions that maintains ecosystem functioning. Different species depend upon one another for food, shelter, and maintenance of suitable environments through complex networks of ecological relationships. This contributes to ecosystem stability and resilience. Consider how certain plant species modify soil chemistry, altering pH levels to create conditions suitable for other plants. Predators regulate prey populations, preventing overgrazing or resource depletion, whilst decomposers break down dead material, recycling nutrients essential for reuse by other organisms. These interconnected relationships create robust ecosystems capable of withstanding environmental changes. When ecosystems possess high biodiversity, they demonstrate greater resilience to disturbances. If one species declines due to disease or environmental change, other species can often compensate by performing similar ecological roles, maintaining ecosystem processes. This acts as ecological insurance, preventing ecosystem collapse when individual species face challenges. However, as biodiversity decreases, ecosystems become increasingly exposed to disruption. Human survival depends fundamentally upon maintaining significant biodiversity levels. We rely on diverse ecosystems for essential services, including food production, medicine development, climate regulation, and oxygen production through photosynthesis. Unfortunately, human activities are reducing biodiversity at unprecedented rates. A primary driver of biodiversity loss is rapid human population growth combined with increasing resource consumption. The global population now exceeds 8 billion people and continues rising, particularly in developing regions. Population graphs reveal dramatic exponential growth, especially during the 20th and 21st centuries. This growth trajectory places enormous pressure on natural resources and ecosystems as humans extract materials necessary for survival and development. When Earth's population remained relatively small, human environmental impacts were typically localized and ecosystems could recover. However, current population levels and consumption patterns create widespread environmental damage. Expanding human populations require increasing amounts of land for housing, agriculture, and industrial development. This expansion occurs primarily through habitat conversion, destroying natural ecosystems and displacing wildlife species. Population growth alone doesn't fully explain increasing environmental pressure. Global improvements in living standards mean individuals consume more resources than previous generations. People increasingly expect comfortable housing, lots of consumer goods, and high tech products. Meeting these expectations requires extracting raw materials, including fossil fuels for plastics production, metals for electronics and construction, and timber for various products. This accelerating resource extraction depletes natural resources faster than regeneration rates. Many critical resources face depletion if current consumption patterns continue. Non-renewable resources like fossil fuels and minerals cannot be replaced on human timescales, whilst renewable resources are being harvested beyond sustainable yields. This overconsumption generates another major environmental challenge: waste management. Increased production and consumption inevitably produce more waste, including hazardous chemicals requiring careful handling. Without proper waste management, pollution accumulates in ecosystems, damaging biodiversity and human health. Water pollution occurs through multiple pathways, severely affecting aquatic biodiversity. Sewage discharge introduces pathogens and substances that disrupt aquatic ecosystems, whilst industrial effluents contain toxic chemicals that bioaccumulate through food chains. Agricultural activities contribute significantly to water pollution through fertilizer runoff and pesticide contamination. These chemicals wash into rivers, lakes, and coastal waters during rainfall, affecting organisms far from application sites. Fertilizer runoff causes eutrophication, triggering algal blooms that deplete dissolved oxygen when decomposing. These conditions create dead zones where most aquatic life cannot survive, dramatically reducing biodiversity. Air pollution presents equally serious threats to biodiversity. Industrial processes and combustion release various atmospheric pollutants, including sulfur dioxide and nitrogen oxides. These pollutants directly harm organisms through respiratory damage and toxic effects. Sulfur dioxide emissions cause acid rain, which damages plant tissues and acidifies soils and water bodies. Acid rain particularly affects forests and freshwater ecosystems, causing species populations to decline and ecosystem degradation. Ground level ozone, formed through reactions between nitrogen oxides and volatile organic compounds, damages plant photosynthesis and reduces agricultural productivity. This affects both wild plant communities and crop systems supporting human populations. Land pollution creates persistent environmental problems affecting terrestrial biodiversity. Landfill sites concentrate household and industrial waste, potentially leaching toxic substances into surrounding soils and groundwater. Improperly managed landfills release methane, contributing to climate change and provide breeding grounds for disease vectors. Agricultural chemicals, including pesticides and herbicides, accumulate in soils, affecting soil organisms essential for nutrient cycling. These chemicals can persist for years, continuously impacting biodiversity and potentially entering food chains. Nuclear waste presents unique challenges, remaining hazardous for thousands of years. Despite underground storage, risks remain of contamination affecting future generations of organisms. These various pollution forms interact together, creating impacts exceeding individual effects. Multiple pollution sources reduce ecosystem resilience, making recovery increasingly difficult even when pollution sources are eliminated. Pollution directly reduces biodiversity through several mechanisms. Sensitive species die when exposed to toxic pollutants, creating gaps in food webs and ecological interactions. And non-deadly effects include reduced reproduction and increased disease sensitivity, gradually depleting populations. Habitat degradation through pollution makes environments uninhabitable for specialized species requiring specific conditions. Other species may increase, but overall species richness and ecosystem complexity decline. Recent decades have witnessed growing recognition of biodiversity's importance and threats it faces. Governments and organizations implement various conservation measures, including protected area establishment, endangered species programs, and habitat restoration projects. International agreements like the Convention on Biological Diversity coordinate global conservation efforts, improved waste management practices, reduce pollution impacts on biodiversity. Recycling programs decrease resource extraction and waste generation, whilst proper hazardous waste disposal prevents environmental contamination. Sustainable resource use strategies, including renewable energy adoption and sustainable farming, help maintain biodiversity whilst meeting human needs. These approaches recognize that human welfare depends upon healthy ecosystems. The future of biodiversity, and ultimately human civilization, depends upon transforming our relationship with nature. This requires fundamental changes in how we produce, consume, and dispose of materials. In this episode of our series, we'll explore how humans use land, investigate environmental impacts of deforestation, explore all the problems caused by this loss of trees, and why peat bogs are environmental superheroes. Humans use absolutely enormous amounts of land for countless different purposes. We need land for building homes and infrastructure, quarrying materials for construction, farming to produce food, and creating landfill sites for waste disposal. Essentially, we've turned Earth's surface into our personal, multi-purpose playground, leaving less room for other organisms. As our population grows and demands more resources, we claim increasingly more land for human activities. This expansion means other species must squeeze into ever smaller spaces. Sometimes our land use has particularly damaging environmental effects. This is especially true when it involves deforestation or the destruction of special habitats like peat bogs, nature's own carbon storing time capsules. Deforestation is the large scale cutting down of forests. While small scale managed tree harvesting can be sustainable, the massive destruction of forests, particularly tropical rainforests, causes serious environmental problems. Why does deforestation happen at such alarming rates? The primary reason is clearing land for agriculture. including cattle ranches and crop cultivation like soya beans, to feed our ever growing global population or crops for biofuel production. Forests are also harvested for timber used in construction, paper production, and various wood products. Unfortunately, deforestation causes multiple environmental problems. First, it dramatically reduces the amount of carbon dioxide removed from the atmosphere through photosynthesis. Trees are nature's carbon vacuum cleaners, efficiently absorbing CO2 and using it to build their wooden structures. Trees lock up carbon in their wood, where it can remain stored for centuries. Think of them as biological carbon prisons. When forests disappear, we lose this crucial carbon capture system that's been working perfectly for millions of years. Additionally, burning trees to clear land releases stored carbon back into the atmosphere as CO2. It's like opening all the prison doors at once. Carbon that was safely locked away suddenly floods back into the atmosphere. Microorganisms decomposing dead wood also release CO2 through respiration. Even after the chainsaws stop, these tiny decomposers continue pumping greenhouse gases into the atmosphere, making deforestation's impact long lasting. Another devastating consequence of deforestation is the loss of biodiversity. Forests, especially tropical rainforests, contain mind boggling numbers of different species, from massive trees to tiny insects. When forests are destroyed, countless species lose their homes, and many face extinction, particularly those adapted to specific forest conditions. Imagine bulldozing an entire city and expecting all residents to just find somewhere else to live. That's essentially what we're doing to forest inhabitants, except they can't simply rent a flat elsewhere. Now let's explore peat bogs, nature's underappreciated carbon storage facilities. Peat bogs are waterlogged wetlands where dead plants don't fully decompose due to oxygen poor conditions. Instead of rotting completely, these plants accumulate over thousands of years, forming thick layers of peat. This process is incredibly important because carbon from dead plants gets stored in peat rather than released into the atmosphere. Peat bogs are essentially Earth's carbon savings accounts, holding more carbon than all the world's forests combined. However, humans drain peat bogs for farmland or dig up peat for fuel and garden compost. We're using peat much faster than it forms, and it's contributing to climate change. Draining peat bogs triggers a carbon catastrophe. Once exposed to air, microorganisms begin decomposing the peat, releasing stored carbon as CO2 through respiration. Burning peat as fuel releases even more CO2, whilst destroying these unique habitats eliminates countless specialized species. It's an environmental double whammy, increasing greenhouse gases whilst reducing biodiversity. Understanding these impacts helps us appreciate why sustainable land use matters enormously. Both forests and peat bogs play irreplaceable roles in regulating Earth's carbon cycle and supporting the incredible diversity of life on our planet. In this episode of our series, we'll explore global warming and its root causes, how greenhouse gases trap solar energy, identify the main types of these greenhouse gases, and finally, the consequences of warming our planet. Global warming is the gradual increase in Earth's average temperature caused by rising atmospheric greenhouse gas levels, and it's affecting weather patterns, sea levels, and ecosystems globally. To understand global warming, we must first grasp Earth's energy balance. Our planet's temperature depends on the balance between incoming solar energy and energy radiated back to space. Atmospheric gases act as Earth's insulating blanket. These gases absorb energy that would normally escape to space and re radiate it in all directions, including back towards Earth's surface. This greenhouse effect keeps our planet approximately 33 degrees Celsius warmer than it would be otherwise, the difference between a habitable world and a frozen wasteland. Problems arise when greenhouse gas concentrations increase significantly beyond natural levels. The primary culprits are carbon dioxide and methane, both increasing sharply due to human activities. Carbon dioxide levels have risen primarily through burning fossil fuels like coal, oil, and natural gas for energy. Deforestation compounds this problem by reducing Earth's capacity to absorb carbon dioxide through photosynthesis. Methane emissions come from livestock farming, rice cultivation, and landfill decomposition. This potent greenhouse gas traps heat far more effectively than CO2, though it persists for less time in the atmosphere. As greenhouse gas concentrations rise, more heat becomes trapped in our atmosphere, driving global temperatures upward. Even seemingly small temperature increases can trigger cascading effects throughout Earth's interconnected systems. The biological consequences of global warming are already becoming apparent worldwide. Rising temperatures melt glaciers and cause thermal expansion of oceans, raising sea levels that threaten coastal habitats where millions of species live. Species distributions are shifting dramatically as organisms track suitable climate conditions. Many wild animals and plants are moving towards the poles or to higher elevations, seeking cooler temperatures. Some species actually benefit from warming conditions. Warm adapted species expand into previously inhospitable regions, sometimes becoming invasive and disrupting existing ecosystems. Mediterranean species, for instance, now thrive in parts of northern Europe that were historically too cold. However, many species face range contractions as suitable habitats shrink. Cold adapted species have nowhere to go when mountains warm or polar regions heat up, facing potential extinction. Migration patterns are changing as animals adjust their movements to new climate realities. Birds arrive earlier at breeding grounds or travel further north, while some migrations are disappearing entirely. These rapid changes threaten global biodiversity significantly. Species unable to adapt quickly enough face local or global extinction, particularly species with narrow ecological requirements. Ecosystem disruption occurs as predator prey relationships, pollination partnerships, and other ecological interactions become mismatched. Understanding global warming's biological impacts helps scientists predict future changes and develop conservation strategies. This knowledge proves essential for protecting vulnerable species and maintaining ecosystem services humans depend upon. In this episode of our series, we'll explore maintaining biodiversity, conservation programs designed to protect ecosystems, strategies to preserve habitats and species, and finally, look at why biodiversity conservation is facing challenges. Maintaining high biodiversity levels is crucial for ecosystem stability and resilience. Biodiversity helps ecosystems remain balanced and recover from disturbances. When biodiversity declines, ecosystems become vulnerable to collapse, potentially triggering cascading effects throughout the natural world. In response to alarming biodiversity losses, scientists and conservationists have developed numerous programs to protect ecosystems and their inhabitants. These initiatives range from small-scale local projects to international conservation efforts. Breeding programs represent one key strategy for preventing species extinctions. These programs serve as genetic insurance policies and involve breeding endangered animals in captivity, ensuring species survival even if wild populations disappear. Individual animals from breeding programs are often released into wild to boost declining populations or re-establish extinct ones. This approach has successfully rescued species like the California condor and Arabian oryx from the brink of extinction. Habitat protection and regeneration programs focus on preserving entire ecosystems rather than individual species. Protecting rare habitats like mangroves, heathlands, and coral reefs safeguards all organisms living within these environments, maintaining both species and ecosystem diversity. Agricultural landscapes also benefit from biodiversity enhancement programs. Many farms now reintroduce hedgerows and field margins around crops, where only single species are grown. Field margins are strips of land around field edges where wildflowers and grasses grow naturally, creating wildlife corridors. These features support far more organisms than could survive in single crop fields alone. Hedgerows provide nesting sites for birds, shelter for small mammals, and food sources for pollinators, significantly increasing agricultural biodiversity. Governments worldwide have introduced regulations to reduce deforestation rates, limiting habitat loss and carbon dioxide emissions. These policies aim to preserve forest ecosystems whilst addressing climate change, tackling two environmental crises simultaneously. Recycling programs reduce pressure on ecosystems by decreasing landfill requirements. When people recycle materials rather than discarding them, less land needs converting to waste disposal sites, preserving natural habitats. Unfortunately, maintaining biodiversity isn't as straightforward as implementing conservation programs. Numerous conflicting pressures complicate conservation efforts, creating difficult choices between environmental protection and human needs. These conflicts often pit short-term economic gains against long-term environmental sustainability. Financial constraints significantly impact conservation efforts. Governments must pay farmers subsidies to maintain hedgerows and field margins, compensating them for reduced crop yields from these areas. Employing wardens to monitor protected areas and enforcing regulations requires substantial funding. Limited budgets mean difficult decisions about which species or habitats receive protection. Economic interests frequently conflict with biodiversity protection goals. Conservation efforts can threaten local livelihoods, particularly in developing regions. Reducing deforestation protects biodiversity but may leave logging workers unemployed, creating social and economic hardship for communities dependent on forest resources. Food security concerns sometimes override biodiversity considerations. Farmers view certain wildlife as pests when they damage crops or livestock, leading to control measures that disrupt food chains and reduce biodiversity. Development pressures constantly challenge conservation efforts. Growing populations require housing, infrastructure, and agricultural land, often at the expense of natural habitats on urban fringes or in developing nations. Finding balance between environmental protection and human needs remains one of our greatest challenges. Success requires cooperation between governments, businesses, communities, and individuals, all working towards sustainable solutions. In this episode of our series, we'll explore what trophic levels are and how they organize feeding relationships, then examine each trophic level in detail, and finally, the role of decomposers in recycling nutrients. Trophic levels are essentially the different stages in a food chain with each level containing organisms that share similar feeding strategies. Trophic levels are numbered sequentially, starting with level 1 at the base of the food chain. Each subsequent level is numbered according to its distance from the primary energy source, also known as producers. Let's examine each trophic level systematically. Trophic level 1 contains producers, organisms that form the foundation of all food chains. These include plants on land and algae in aquatic environments, both capable of manufacturing their own food through photosynthesis. Producers harness solar energy to convert carbon dioxide and water into glucose. They provide themselves with energy whilst building complex molecules for growth and are essentially biological solar panels, converting light energy into chemical energy that powers entire ecosystems. Without producers capturing energy from the sun, no other life could exist on Earth. They provide the energy base upon which all other trophic levels depend. Trophic level 2 contains primary consumers, the herbivores. These organisms feed exclusively on producers, obtaining energy by digesting plant material. Examples include cows munching grass, caterpillars devouring leaves, and tiny organisms such as zooplankton consuming algae in oceans. Primary consumers are called primary because they're the first consumers in the food chain, directly accessing energy stored by producers. They've evolved specialized digestive systems to break down tough plant materials like cellulose, extracting nutrients that carnivores couldn't access directly. Trophic level 3 contains secondary consumers, carnivores that prey upon herbivores. These predators, including foxes hunting rabbits, birds catching caterpillars, and small fish eating zooplankton, obtain energy by consuming primary consumers. The energy these carnivores obtain originated from sunlight captured by plants, then passed through herbivores. With each transfer, substantial energy is lost through respiration, movement, and heat production, making less energy available at higher levels. Trophic level 4 contains tertiary consumers that eat other carnivores. These high level predators, such as eagles catching snakes, polar bears hunting seals, or sharks consuming smaller predatory fish, sit near the top of food chains. They require large territories or ranges because the available energy at this level supports fewer individuals. Some carnivores have no natural predators except humans and occupy the highest trophic level in their ecosystems. These apex predators, like lions, orcas, and crocodiles, regulate prey populations and maintain ecosystem balance through top-down control. Food chains rarely extend beyond 4 trophic levels because energy losses at each transfer are substantial. Approximately 90% of energy is lost between levels through respiration, movement, and heat production, leaving insufficient energy to support additional levels. Decomposers operate outside the traditional trophic level structure but remain essential. Bacteria and fungi decompose dead organisms from all trophic levels, breaking down complex organic matter into simple compounds. They secrete digestive enzymes into their surroundings, breaking down proteins, carbohydrates, and fats into small, soluble molecules. These molecules then diffuse into decomposer cells, providing them with nutrients while simultaneously releasing minerals back into soil or water. This recycling process ensures nutrients remain available for producers, completing the nutrient cycle. Without decomposers, dead organisms would accumulate, locking away nutrients indefinitely. Ecosystems would eventually collapse as producers exhausted available minerals, demonstrating decomposers' critical ecological role. Understanding trophic levels reveals how energy flows through ecosystems and illustrates organisms' interdependence. Each level depends on those below it for energy, creating complex webs of feeding relationships. In this brief episode, we will discuss the pyramids of biomass and what determines their structure. Pyramids of biomass show the total mass of living material at each trophic level in a food chain. Each horizontal bar relates to the combined biomass of all organisms at that particular feeding level. As you move further up in trophic levels in a food chain, there's less and less energy and biomass at each stage. This pattern happens because energy is lost at each transfer between the trophic levels, supporting fewer organisms and less total mass as you move up. This energy loss explains why food chains rarely exceed 4 levels. With such inefficient transfers, there's simply not enough energy remaining to support additional trophic levels beyond the fourth or fifth. Consider the following woodland food chain. A simplified example might be that 1,000 plants might support 100 caterpillars, which sustain 10 birds, and finally, those support a single eagle. The total mass of plants exceeds the combined mass of caterpillars, and this pattern continues. Counting individual organisms can be deceptive. Imagine 100 fleas feeding on the eagle. Though greater in numbers, the fleas' combined biomass remains tiny compared to their host. Understanding pyramids of biomass helps ecologists assess ecosystem health and stability. Changes in biomass at one level can predict impacts throughout the food chain, making these diagrams valuable for conservation planning. These pyramids also explain why large predators require extensive territories. Supporting a single top predator requires enormous amounts of producer biomass at the pyramid's base. This episode of our series builds on the previous one. We'll quickly revise what biomass is, then look at why biomass is lost at each trophic level, and finally, how to calculate biomass transfer efficiency. Consider the journey of energy through a simple food chain. Grass grows using sunlight, cattle eat grass for growth, and humans consume beef. At each step, substantial energy disappears. The cattle use most of their consumed biomass for their own survival, movement, and bodily functions. Additionally, not everything cattle eat can be digested and absorbed. This illustrates the fundamental reality of biomass transfer. It's an inherently wasteful process with massive losses at every stage. Biomass is the total mass of living material in organisms. This biomass transfers through food chains when organisms consume other organisms. However, biomass transfer between trophic levels is startlingly inefficient, with only about 10% passing to the next level. This means 90% of biomass is lost at each transfer, a staggering amount of waste. Why does such dramatic biomass loss occur between trophic levels? Multiple biological processes contribute to this inefficiency, each representing energy that cannot pass up the food chain. First, organisms rarely consume every part of their prey or food source. Herbivores might avoid woody stems, thorns, or roots, whilst carnivores often leave bones, fur, or feathers uneaten. These inedible portions represent biomass that never enters the consumer's body. Even when dealing with apparently edible material, structural components like cellulose in plants prove difficult for many animals to digest. The digestive system cannot break down everything consumed, and undigested material passes through as feces. This waste exits the food chain entirely, returning nutrients to decomposers rather than passing to higher trophic levels. Third, Absorbed biomass gets metabolized for various life processes, producing waste products. Respiration uses glucose for energy, releasing carbon dioxide and water as byproducts that cannot be recaptured by the organism. Animals use substantial energy for movement, maintaining body temperature, and other metabolic activities. Warm-blooded animals particularly lose enormous amounts of energy as heat, which dissipates into the environment. Protein metabolism produces nitrogen-based waste excreted as urea in urine. This represents another pathway through which biomass leaves the food chain, carrying away materials that required energy to process. We can calculate biomass transfer efficiency using a straightforward formula. Consider a pond ecosystem with algae, water fleas, small fish, and large fish. The numbers shown represent biomass available at each trophic level. To calculate biomass loss between levels, subtract the biomass at the higher level from that at the lower level. Between algae and water fleas, 364 kilograms is lost. Transfer efficiency equals biomass transferred divided by biomass available multiplied by 100. For algae to water fleas, there is a 9% transfer efficiency. This calculation reveals that only 9% of algal biomass transfers to water fleas. Similar calculations for other levels would show comparably low efficiencies, typically ranging from 5% to 20%. These low efficiencies explain fundamental ecological patterns. Food chains rarely exceed 4 levels because insufficient energy remains to support additional trophic levels. By the fourth level, only about 0.01% of the original solar energy remains available. This also explains why organism numbers generally decrease at higher trophic levels. Supporting 1 kilogram of large fish requires thousands of kilograms of algae at the base of the food chain. Apex predators like eagles or sharks are necessarily rare because the energy available at their trophic level can support only small populations. Their rarity isn't accidental, but mathematically inevitable given biomass transfer inefficiencies. Understanding biomass transfer reveals why protecting primary producers is crucial for ecosystem stability. Damage to producer populations cascades up food chains, with effects magnified by the 90% loss at each level. In this episode of our series, we're covering a variety of topics. We'll begin by understanding food security and the complex factors threatening it worldwide, how overfishing depletes ocean resources, modern farming techniques, and finally, we'll consider the ethical implications of intensive farming. Food security means having reliable access to sufficient, safe, and nutritious food for an active, healthy life. It encompasses not just food availability, but also accessibility, affordability, and nutritional adequacy. A population can have food security even if they import much of their food, provided supply chains remain stable and people can afford adequate nutrition. Unfortunately, numerous factors threaten food security globally, creating vulnerabilities that affect billions of people. Understanding these threats is essential for developing effective solutions to ensure everyone has access to adequate nutrition. Population growth represents a fundamental challenge to food security, particularly in developing nations where birth rates remain high. The global population is expanding rapidly, requiring increased food production from essentially the same agricultural land area. This creates mounting pressure on agricultural systems already stretched to capacity. Changing dietary patterns in developed countries compound this challenge through increased demand for resource intensive foods. As incomes rise, populations typically consume more meat, dairy, and processed foods, which require far more resources to produce than plant-based diets. Additionally, globalization means food produced in developing countries increasingly gets exported to wealthier nations. This international trade can improve farmers' incomes, but may reduce local food availability, creating food insecurity even in agriculturally productive regions. Agricultural production faces constant threats from pests and pathogens that can devastate crops and livestock. New diseases emerge regularly through mutation or geographic spread, whilst climate change enables pests to expand into previously inhospitable regions. A single outbreak can destroy entire harvests, as seen with locust swarms in East Africa or African swine fever in Asia. These biological threats can trigger widespread famine in regions with limited resources for pest control or disease management. Environmental changes increasingly disrupt traditional farming patterns worldwide. Droughts, floods, and extreme weather events are becoming more frequent and severe due to climate change. When monsoons fail or unexpected frosts occur, millions can face food shortages as crops fail and livestock perish. Rising temperatures alter growing seasons and shift suitable agricultural zones, forcing farmers to adapt or abandon traditional crops. Coastal agricultural areas face saltwater intrusion from rising sea levels, reducing productive land area. The high costs of modern farming create barriers to food production in many regions. Machinery, fertilizers, pesticides, and irrigation systems require a large investment that small scale farmers often cannot afford. This economic barrier means many communities cannot increase production despite having suitable land and climate. Without access to modern techniques, they remain vulnerable to crop failures and cannot produce surpluses for market sale. Armed conflicts devastate food security by disrupting production, distribution, and access. Wars destroy farming infrastructure and relocate farming populations, thereby preventing cultivation even where environmental conditions remain suitable. Conflict zones often experience severe food shortages as supply chains collapse and humanitarian access becomes restricted. Water conflicts particularly threaten food security, as farming consumes approximately 70% of global fresh water. Disputes over river access or groundwater extraction can leave entire regions unable to irrigate crops. Overfishing represents another critical threat to global food security, particularly for coastal populations dependent on marine protein. Fish stocks worldwide are declining due to industrial fishing methods that harvest fish faster than populations can reproduce. Many commercially important species face potential collapse. North Atlantic cod exemplifies this crisis, with populations crashing to near extinction levels despite once supporting massive fisheries. Similar declines affect tuna, sharks, and numerous other species that require decades to reach reproductive maturity. Overfishing disrupts entire marine ecosystems by removing key predators or prey species. This can trigger cascading effects throughout ocean food webs, potentially causing irreversible ecological changes. Sustainable fishing practices offer solutions by maintaining fish stocks at levels supporting continued reproduction. This requires careful management based on scientific assessment of population changes and ecosystem interactions. Fishing quotas establish limits on catch quantities and fish sizes for specific areas and seasons. These regulations prevent overharvesting whilst allowing younger fish to reach breeding age before capture. Net design modifications significantly reduce unsustainable fishing impacts. Larger mesh sizes allow juvenile or younger fish to escape, ensuring they survive to reproduce before entering the fishery. This simple change can dramatically improve population sustainability whilst maintaining commercial viability, reducing bycatch, the unintended capture of non-target species, protects ecosystem diversity. Modified nets and fishing techniques can minimize capture of dolphins, turtles, and other vulnerable species often killed in fishing operations. Modern farming techniques aim to increase production efficiency by controlling energy transfer within agricultural systems. By minimizing energy losses to the environment, more energy becomes available for producing food. Intensive animal farming restricts animal movement within controlled environments, reducing energy spent on movement. Temperature regulation in these facilities prevents energy loss through thermoregulation, allowing more consumed energy to support growth. These efficiency gains come with significant ethical considerations. Animals in intensive systems often live in cramped conditions. Chickens may never see daylight, whilst pigs cannot express natural behaviors like nest building. Disease spreads rapidly in crowded conditions, necessitating routine antibiotic use that contributes to antimicrobial resistance. This creates public health risks beyond immediate animal welfare concerns. High protein feeds accelerate growth but may cause skeletal problems in rapidly growing animals. Broiler chickens often cannot support their own weight by slaughter age, experiencing pain and mobility issues. Fish farming presents similar challenges, with salmon kept in sea cages at densities far exceeding natural conditions. Parasites and diseases spread quickly, requiring chemical treatments that can affect wild populations. Society must balance productivity needs against ethical responsibilities. Some argue that intensive farming is necessary to feed growing populations affordably. They emphasize that without these methods, food prices would rise beyond many people's means, creating worse welfare outcomes overall. Others contend that animal suffering cannot be justified by economic efficiency. They advocate for higher welfare standards even if this increases costs, arguing that society should prioritize compassion over cheap food. Alternative approaches seek middle ground between intensive production and extensive systems. These include free range farming and rotational grazing, which improves welfare whilst maintaining reasonable productivity. Precision farming uses technology to optimize resource use without animal restriction. GPS guided machinery, soil sensors, and drone monitoring enable farmers to apply exactly what crops need, reducing waste whilst maintaining yields. International cooperation remains essential for addressing food security challenges as fish populations migrate across national boundaries, making conservation efforts by individual countries ineffective. Individual choices also influence food system sustainability through purchasing decisions and consumption patterns. Reducing food waste, choosing sustainably produced foods, and moderating meat consumption can collectively reduce pressure on agricultural systems. As populations continue growing and climate change intensifies, achieving food security whilst preserving environmental resources becomes increasingly challenging. Success requires integrating technological innovation, policy reform, and behavioral change across all levels of society. In this episode of our series, we'll take a look at what biotechnology is, mycoprotein, genetically engineered bacteria, and finally, we'll explore genetically modified crops. Biotechnology involves manipulating living organisms and biological processes to create useful products. Modern biotechnology enables scientists to culture vast quantities of microorganisms under precisely controlled industrial conditions. These organisms, grown in large fermentation vessels, produce valuable substances ranging from food ingredients to life-saving medicines. Let's examine mycoprotein, a protein-rich food product derived from fungi that's becoming increasingly important as a sustainable meat alternative. The most well-known example uses Fusarium venenatum, grown on glucose-rich nutrients in aerobic conditions within temperature-controlled fermentation tanks. The fungus converts simple sugars into protein-rich biomass remarkably efficiently. After fermentation, the fungal culture undergoes heat treatment to reduce harmful RNA content. Then filtration and further alterations create products with meat-like texture. The resulting mycoprotein contains approximately 45% protein by dry weight, plus essential amino acids and dietary fiber. This production method offers substantial environmental advantages over conventional meat production. Mycoprotein requires 90% less land and water than beef production, whilst generating 90% fewer greenhouse gas emissions. For every kilogram of mycoprotein produced, only 1.5 kilograms of glucose are required, demonstrating exceptional feed conversion efficiency. Compare this to beef production, which requires approximately 10 kilograms of feed per kilogram of meat. Biotechnology has also revolutionized medicine through microbial production of human proteins. Consider human insulin production, which transformed diabetes treatment worldwide. Before genetic engineering, insulin was extracted from pig and cattle pancreases, causing allergic reactions in some patients and requiring enormous numbers of animals. Scientists now insert the human insulin gene into bacteria, typically E. coli, which then produce human identical insulin. The bacteria multiply rapidly in fermentation tanks, creating vast quantities of insulin that's purified and formulated into medicine. This process ensures consistent supply, reduces production costs, and eliminates animal-derived contaminants. Similar techniques produce human growth hormone for treating growth disorders, clotting factors for hemophilia, and numerous other therapeutic proteins. The precision of genetic engineering enables targeted solutions to specific medical needs. Genetically modified crops represent another significant biotechnology application addressing global food security. Scientists can introduce specific traits like drought resistance, pest resistance, or enhanced nutrition directly into crop genomes. Golden rice exemplifies nutritional biofortification containing genes that produce beta carotene, a vitamin A precursor. This addresses vitamin A deficiency affecting millions in developing countries, potentially preventing blindness and reducing child mortality. Bt cotton and Bt maize contain genes from Bacillus thuringiensis bacteria, producing proteins toxic to specific insect pests but harmless to humans. Farmers growing these crops report 30 to 80% reductions in pesticide applications, improving both environmental outcomes and farmer health. However, GM crops generate significant controversy and debate worldwide. Multiple concerns exist regarding their environmental, social, and economic impacts. Critics argue that hunger results primarily from poverty and inequality rather than insufficient food production. They contend that addressing economic disparities would more effectively tackle lack of nutrition than technological solutions. Environmental concerns include potential gene flow to wild relatives, creating superweeds resistant to herbicides. There's also worry about impacts on non target organisms like monarch butterflies affected by Bt crop pollen. Additionally, GM crops cannot overcome fundamental agricultural challenges like severely degraded soils or extreme weather. Even engineered drought resistance has limits when facing prolonged water scarcity or degraded land. Corporate control over GM seeds raises socioeconomic concerns. Large biotechnology companies own patents on GM varieties, requiring farmers to purchase new seeds annually rather than saving them. This creates dependency and can increase farming costs, particularly affecting farmers in developing nations. Some worry that biotechnology diverts attention from simpler, less expensive approaches that might provide more sustainable solutions. Traditional breeding, crop rotation, and integrated pest management could address many agricultural challenges without genetic modification. Despite controversies, biotechnology continues advancing rapidly, offering tools to address food security, nutrition, and health challenges. Success requires careful regulation, transparent communication, and consideration of both benefits and risks.